concerning the Asturiana de los Valles breed, and which has been possible to do because we have gene banks available. Gene banks in the very amplitude way. Well, I would like to acknowledge before I start my colleagues in the MAGE project, who have made possible to model all this information, so that I'm not going to enter in details, but there's one of them, Bertrand Servin, is in the room, I think, and if you have questions at the end, he can answer. Okay, I'm going to initiate this talk very simply with very basic knowledge about artificial selection. And we know that humans have driven selection since domestication, and this selection has left very variable signs in the genome. So when selection is made, artificial selection is produced by human, the selection tends to leave very variable signs and patterns in the genome, and not only at the selected loci, but also in the loci which are around, all the nucleotides that are around, in a sort of hitchhiking process. So allele frequencies are going to change when the loci gene is selected, but also the allele frequencies of the surrounding nucleotides and loci. And this is what we call signatures of selection. So these signatures of selection are very important to identify really the loci which are subjected to selection and understand complex traits which are difficult to apprehend. In the last years, we have assisted to high increase of genotyping, mostly of single nucleotide polymorphisms, SNPs, that are now very cheap to produce. We have here in the presentation was Affymetrix or Illumina chips, which are really very low cost, and which allow, by genome-wide association studies, to detect the regions which have signatures of selection. Many statistical methods have been developed in the last years, and we can resume them and summarize them in three groups. Mostly measures based on allele frequencies changes, within population measures based on extended haplotypes, homozygosity, or measures based on differentiation between and within species. This is not the place to talk about these different methods, but just to say that they are there, and we will use one of those also. So detection of selection signature can increase the understanding and the evolution of biology underlying a given phenotype, and may provide tools to increase the efficiency of selection. This is the tool. Past positive selection affects genetic diversity and can be detected from present day samples. So usually what we do is to analyze present day animals or samples and to detect the past positive selection patterns, which can be very old. The thing is that with those samples, we are not always allowed to say if it's an adaptive trait. We are not allowed really to say which was the onset of this selection and what was the intensity of this selection. So we only know these signatures of selection, and we cannot go further. But now with this project of MAGE, we are really aware that we have gene banks. Not only semen, we have also DNA samples from very different years. Recent years, but in the last 30, 50 years, we have samples. So these samples are able to be analyzed to detect recent selection, and it will be possible with them to annotate signatures of historical selection. Well, this is the question that we ask. So for this purpose, a method has been developed in this project. It was published just in December, I think. So you have here the publication. This software is, is trying to see for the evolution of the allele frequencies in different time series. And they use something, it's not my model, so that's why I'm not really talking very well about it. It's following a kind of Wright-Fisher model, but it tries to approximate transition probabilities between two time points. So this is what we are going to use. The case study that we have applied here is the Asturiana de los Valles samples. The Asturiana de los Valles is a bovine breed, which a beef bovine breed, located mostly in the north of Spain, where you have the here, and is characterized by two signs. One is this very special type of production system. In Asturiana de Valles, we still the breeders are using the very old, extensive system, which is in the mountains. You have it on the right. Well, I don't have the pointer. On the right, you can see most of the animals are kept indoors during the winter and are in a kind of feral way left in the hills during 4 or 5 months a year. So there they are faced to predators. There are wolves over there. There are bears. Well, in the last years, there is an intensive production also of Asturiana de los Valles in improved fields. And in the last years also, there is a replacement of the dairy farms with Asturiana de los Valles. This is one characteristic because it's quite specific. The other characteristic is the double muscling. In this breed, we can find a very high frequency of one deletion of one mutated allele of the myostatin, which gives the double muscling. And here, this is a study which has been done in another project. We can see the phenotypes, how it produces or it alters completely phenotypes for beef characteristics. And what you can see, the Asturiana de los Valles is the red line. So the red line, what you can feel is that there is very little fat in the Asturiana de los Valles carcasses, but this fat has a very high percentage of polyunsaturated fat, lipids. The sensory phenotype is very good, it's very high range, and the carcass production is also quite high. You can see this red line. Compared with other beef breeds like Aberdeen Angus, Limousin, I think there's over there, I can't see here, Limousin, Holstein or Charolais. Okay, so this breed has in gene banks samples which are dated from 1980 until 2000, well, until now, but we took for this work until 2015. So 35 years of evolution, which we are going to look for. Okay, we sampled 153 samples, which came from different projects. So some of the samples were genotyped with the Illumina medium density chip. Others were sampled with the high density chip Illumina also. And 15 were sampled and were sequenced, whole genome sequenced in the MAGE project. So we had the complete genome of these 15 animals. Here you see how according to the birth dates, the samples are according to the birth dates and to the type of genotyping. We covered, of course, the MAGE animals are in the last 2 generations, and most of the first generations are covered by the Affymetrix medium density chip. Okay, well, we used all this information, we made some SNP calling, we erased some missing calling SNPs. So at the end, we had these numbers, more than 14 million SNPs detected by the whole sequencing done in MAGE, and in common, about 35,000 SNPs. On the right side of the slide, you can see the genetic diversity of the markers as a summary, and we can in a PCA analysis, and you can see that there is no detectable effect on the technology, and this was good for us because we merged. Sorry, I think I'm gone. No, it's okay. We merged all the data. So what we did is, with all these 35 years evolution of samples, we tried to detect a generation time of 4 years. 4 years is slightly less than what is the real generation interval in Estudiar los Valles, but there is a high correlation between 4 years interval and 5 years of interval, so this is okay for the study. We tried to adjust for the hypothesis of, also under the model of Wright and Fisher model. We tried to eliminate all the animals which were inbred above 1.07 and with the related animals which had a coancestry above 0.1. So at the end, well, we had 117 animals which were distributed by birth date, and we had 9 generations with this number of samples. Okay, so what we did is to use a model, a statistical model, which allows us to detect old selection. So we chose NSL statistic to detect the signatures of historical selected only in the animals which were analyzed in the IMAGE project. This statistical model detects long haplotypes that have very high frequency, and having this sequencing we had much more SNPs to study, so the signals were much higher. Much higher detection power. The only problem with this model is that no proper p-value is associated to a given NSL score because we don't really know which is the distribution in this model. So p-values are not very indicative of nothing. So we use an outlier approach, and we really use the very high signals that come out. We also used our new model, time series model, which exploits the evolution of allele frequencies in a population along different sampling times. In this way, it is under the Wright and Fisher model, or very similar, with a sort of modification. I don't give us the details, but it approximates the transition probabilities between 2 points in the evolution of the allele frequencies. So you have again the article here, the publication, if you want to have a look. Okay, so at the first we made the analysis with the present time data, with the individuals which were from generation 8 and 9, and which were sequenced, whole genome sequenced. But we only used first the common SNPs, so the 35,000 SNPs which were common with the other chips. And we found these 5 regions with very strong signals in chromosome 2, in chromosome 6, in chromosome 10, and chromosome 25. So here in this table, you can see the candidate genes which are included in the regions. So in chromosome 2, which is the chromosome where myostatin locus is included, we can see collagen candidate genes which are in the process of hitchhiking, which are very near from the myostatin gene, very close. I must say that the mutated allele of the Ashton-Los-Bayes, which is in a very high frequency, is a 11-pair deletion. So this is not normally included in the Illumina or in the Affymetrix chip. So we are not going to find the SNP because it's a deletion, but we find the surrounding loci which are in the region. Okay, there is also FLRT2, which is in the chromosome 10, which was found to be included in or associated with calf birth weight in the Holstein breed, and which has something to do with embryonic development and with craniofacial development, and is interesting. It's interesting to find here in Asturias-Los-Valles. Okay, I'll go further. What we did in the second step was to use this present time data, the animals analyzed in the image of generation 8 and 9, but with all the SNPs that were found in the whole genome sequencing. So more than 15 million SNPs. And so the detection power was much increased. Of course, we have much nicer results. And we find these regions which are very high signal. Again, the chromosome 2. Now in chromosome 4, in chromosome 10 again, with the same loci, the same candidate gene, FLRT2, which is associated to birth weight, and in chromosome 15. Okay, I'm going to talk about this candidate region a bit further. Okay, in the third step, what we did was analyze with this time series model. The time series model uses all the generations, all the 35 years in intervals of 4 years, and we detected 5 significant regions. One is in chromosome 10, which is again where FLRT2 is included, but we don't find this loci in the region, probably because the detection power is lower. But all the genes that are detected here in chromosome 10, 13, 17, and 22 are all related to carcass and meat traits and also to milk traits. F1P1, which is included in chromosome 17. Okay, so I'm going to try to make a comparison between the results. If we look at chromosome 2, which is an expected result, because Asturias-Los-Valles has a frequency of this allele of 80% frequency now at the moment. So then the region which arise from this study is the region including the myostatin, but what we see is other genes because of this fact that this mutated allele is an 11-pair deletion. This 11-pair deletion was described in 1997, and on the right you have a photograph of a bull, of an Asturias-Los-Valles bull, who is double muscled. So what we see here are all the other genes which are hitchhiked and which are in the region. Okay, chromosome 4. In chromosome 4, what we find is a cluster of genes of the GMAP family. It's a GTPase immunity family members, and they have been implicated in humans in immunity processes, and also they have been associated in bovine. In the last years, there is a publication in 2017 on feed efficiency with one or more of these genes in cluster, and with metabolism of lipids and sugars and proteins, so mostly feed efficiency. This is something that in Asturias-Los-Valles is quite interesting to know that it exists. In chromosome 10, we didn't find anything with the time series model, so this indicates that chromosome 2, the myostatin, and chromosome 10 results FLRT2, are old selections. I mean, when the samples of the time series started, this selection was already there. So it is old events of selection which were there, and we can annotate them quite easily like that. So we have these different genes which are included in morphology, in bone morphogenetic protein pathways, cell growth, proliferation, etc. And finally, in the chromosome 15, we have a group of olfactory receptors, which is quite funny to find, and we can perhaps link them to better capacity of feeding and also to avoid predation, because this breed has always been in extensive conditions, as I told at the beginning. And in the chromosome 17, we have also genes which are associated with myogenesis or milk traits. I'm ending now. Just a comparison between the two approaches. If we take the very high signals we have found in the NSL statistics and in the time series, NSL on the left and time series model, and we look all over the time, we can see that those of old selection have no or very slight variation. That means they are old selection, really, the 5 on the left, and the ones since 1980 have a lot of variation. On the right, we can see, so they are due to selection, which is which is very not very old. Okay, just some take-home comments. Time series approach complements the standard present time sampling approach. So we have found new loci which are interesting. Relevant candidate genes identified with the two approaches relate to meat, milk production, immunity, olfaction, which is a new perspective that we have to see. And really time series material stored in gene banks. Gene banks are really a very useful reservoir of samples, represent a key resource for the understanding of breed history and the detection of relevant functional genes and variants. Okay, thank you.
Thank you. There's time for a few short questions. Amber?
In relation to the selection signals that you have detected in the olfactory receptors, what type of selection was it? Was it balancing selection?
This is only identification of genomic regions. And what we look is what are the loci, the candidate genes, which are in the regions. Now we have to develop another study to see what is about all this information. So this is not an information that we still have. So it's a further work.
Just one management question. You said that this breed is kept outside on pastures in a far away, and in the same time it has a certain frequency of the gene that is inducing double muscles. So how does it work? How will calvings look like when you have cows that may have cows with this gene, and that create any problems?
No problem. I mean, when you compare with, for example, the Belgian blue breed, which has a lot of problems of dystocia, Asturias de los Valles has a very, very low dystocia percentage. It's I think it's 97% of the cows have a calf ease, a normal calf ease. So there's, yes, and this is a good thing because this this production system does not allow to have them very near. So it's one of the characteristics also of the Asturias de los Valles breeds.
Okay, Susanna, thank you very much. Another example what we're going to show now in the next presentation by Paolo is what we can do with molecular data and gene bank collections and breeds to find interesting genes for that.
Okay, I'm very bad in the. I can see. Please. Thank you, Richard.
Okay, and I think it was already open, huh? No.
That one. Here. This one.
This one?
Yeah.
I can't see.
Yeah.
It's
Difficult to see. Thank you.
Yes. Okay, go ahead.
Thank you very much. Thank you. So, good afternoon to everybody. I'm talking about adaptation to environment by landscape genomic. I would say that a better title would be trying to decipher the adaptation instead of deciphering the adaptation. So this was part of the work package 4 of MAGE that was led by Richard Kreuzmann. And the main people involved in this task were the EPFL, Università Cattolica, where I work, and IRA Morocco. So the idea started looking at where sheep in particular was domesticated in the Fertile Crescent, and after domestication, it colonized the world, and during this colonization, it adapted to a very wide range of environment. Of course, the story is more complex, but this is the basic process that led to adaptation to very different environment in terms of humidity, temperatures, day length, light length, and so on. So in why adaptation, it's very important probably right now to try to understand it because of rapid climate change. So this makes the traditional breeding probably not fast enough if we have to really select for better adapted animals. And maybe molecular genetics can, genome can help in this task, maybe. So the approach we used is this landscape genomics is a new idea that was brought by Stefan Just, I would say, quite a long time ago during the Econogene project. And with the help, it was developed also with the help of Mike Bruford. And the idea is to join genomic data and environmental data to try to identify loci and genes that are linked to adaptation to specific environment. Of course, at that time, we had only a bunch of markers, and this task was, the idea was good, but the task was not really successful. So the idea of landscape genomic basically is that is to identify genotypes instead of alleles that are associated to differential climates. So here the homozygous B is more frequent in cold climate and homozygote A in hot climate. So to accomplish this task, first the European, the study area was divided in grids and then clustered by using bioclimatic variables and elevation in environmental clusters. So basically, this was done by Oliver Selmoni at APFL. He identified 8 main clusters that are here represented and 32 subclusters. These bioclimatic variables are a range of variables that are means of temperatures or isothermality. It means how during the year the temperature is varying and so on and so forth. So in parallel, in Piacenza, we were collecting samples and data. And basically, we were collecting samples from already genotype data, and this was a contribution basically of all the partners of IMAGE. And then we had also the funding. The original idea is to use only whole genome sequences on 100 animals. Basically, because of the decrease of the cost of sequencing, we could analyze HT with the 600,000 sheep, more than 700 animals. And at the end, our study sampling was included more than 14,000 samples. 1,400, samples, and among these, 100 were completely sequenced. And this is the sampling area. Well, there's much more samples here than in Morocco. The difference is that in Morocco, they are very well, this come from the NextGen project, and they were very well spread over the area, while here there's a lot of overlapping between individual animals. So they were assigned to the different climatic clusters, depending, this is continental humid, Mediterranean dry, continental cold, arid, desert, Atlantic dry, Atlantic wet, and alpine. These are the 8 clusters. And basically, at the end, after the cleaning stage, we had more or less cleaning for LD, for the number of animals in a breed. We had about 168,000 SNPs on 11,000 individuals from 12 countries and 77 breeds. And well, after running the analysis that we'll see in a few moments, we collected candidate genes in a very narrow range compared to the associated SNP, so a narrow area. This because the linkage decay, the disequilibrium decay in looking all the samples we had was very steep. And this was compared to normal analysis. GWAS analysis is clearly an advantage because you don't have to include many genes or many variants in your candidate genes and variants. About the whole genome sequences at the end, well, there was a 2-step selection of animals to be whole sequenced. First, there was one animal per 53 breeds, and then on the basis of a preliminary analysis of association with environmental variables, we selected the extreme values, animals that were in extreme values of associated environmental variables, having opposite homozygote genotypes for promising candidate genes. Okay, so at the end, we increased the number of animals to 100, but the number of breeds only to 1. There was a lot of data, 14 billion reads with more than 4,000 gigabases of data. The average depth was 12 times. We made a number of exploratory analyses. I only showed this one. This is a principal component analysis. These are the breeds, but the color corresponds to the environmental clusters. So it is clear that there is a structure in the data, both in terms of breed effect and also in terms of environmental association of breed, environmental rearing of the different breeds. So it was necessary to correct for population structure to identify candidate genes. So we did that with landscape genomics. That experiment was running at EPFL. Basically, the landscape genomic is looking at the probability of the presence of a genotype on the basis of selective, in a certain area on the basis of selective pressure. This selective pressure, in this case, is the environmental variable. So the model is this, is the likelihood of having the genotype in the sampling side, considering the variable that account for population structure. So the scores of the principal component analysis, basically, and the same likelihood in which also the environmental variable is added. So, and then there is a likelihood ratio test. Is the environmental variable contributed something? If this is so, there will be a significant likelihood ratio test. Okay. So the variables that were tested were 51. So the bioclimatic variables we have seen before, then the average minimum, average maximum monthly temperatures, monthly precipitations, and altitude. So to identify how to correct, in this case, Sambada is correcting for the between breed variation. Okay, so basically it was done a PCA, a principal component analysis, and then was done a discriminant analysis on the principal component scores. And basically the selection of the number of genetic clusters identified by this kind of analysis was 8. So looking at the 7 eigenvalues of the discriminant analysis, the scores of these 7 dimensions were used to correct for population structure before going for association with environmental variables. So at the end, there was correction for multiple testing, and there were at the end 195 genes, okay, that in the close interval from the marker that had a gene name. So we in this moment focus only to transcript or gene predicted that had a gene name. And we made a gene ontology analysis using G-profiler. So of course, looking at molecular function, you have you're very high in the gene ontology level. You have protein binding and molecular function. If you go down to the gene ontology classification to biological processes, you start having some information. You have some on metabolism, homeostasis, cellular processes, and regulation. And then there is also one term here that is anatomical structure development, so the shape of the animal. Looking at the cell compartment, they are everywhere, so in the cytoplasm and in the organelle. And it is interesting because G-profiles is also giving a link to human phenotypes. This means that, of course, these are, how do you say, clinical phenotypes, so some abnormalities. But it is interesting to see that there is a classification, there's morphology, there's behavior, so in the brain, cardiovascular, so the heart and the cardiovascular system, the digestive system, and some in the immunitary system. So we didn't stop here. We wanted to have a few genes to study in more detail, so we ran other 2 different selection signature approaches. One is PC-adapt, that has a complete different approach because PC-adapt finds out markers that outliers compared to the population structure. So it is an outlier marker within the breeds, basically, that are identified. I don't want to go into much details here, but takes into account population structure. There's no need to group animals into populations, so it uses the clusters found in the principal component analysis to find then outliers. And in this way, it also handles admixed individuals. To correct for population structure, what is usually done is a principal component analysis, and then you decide how many dimensions used to, the scores of how many dimensions to use for correcting. In this case, according to the Cratten's rule, Elia used K equal to 12 to correct. And this PC-adapt approach identifies 643 SNPs and 211 genes. And then the last that we applied is the classic FST with no, quick and dirty, no population structure correction. In this case, Mario did it, and we found 101 genes. Then we put together all the data, and we found basically quite a low number of genes that to concentrate on. So 11 were confirmed by all of them, and 16 and 5 were confirmed, identified in some data, and at least in another one of the approaches. So, and these are the 11 genes. Some, are so close, like this FAM 184B and NCAPG, that have been identified basically by the same marker, and also in this case, the RXFP2 and this one. And they have been associated by some data to this kind of environmental variables. So now arriving to the candidate gene is easy. Now going one step further to understand why they were associated to environmental variables and why they were under selection is much more tricky, okay? Anyway, these were precipitation, and then the bio are here. Precipitation in the wettest month, in the driest month, in the driest quarter, temperature annual range, and isothermality, okay? We will see some more detail in the next slide. So these are the 11 genes, and an effort to find the function of the gene and the association. What we found is that basically all of them have been pinpointed as under selection in different species. In different species means cattle, but sometimes means buffalo, means goat, and sometimes also humans and mouse. So there is a multiple evidence of these genes to have been under selection or to have an adaptive value somehow. Classifying them, there's many on morphology, then metabolism, adaptation, and disease. Some have a double arrow here because, for instance, this RXFP2 has been identified for horn morphology in sheep, but also has been reported for important for climate adaptation and desert adaptation. We'll see that in this case, horn morphology is the correct answer, and probably this indicates that we have to think about correcting not only for population structure, but also for the kind of management of breeders, of the breeds, okay? We find some that are quite familiar with. So ABCG2 is very important and very famous for association with milky decomposition in sheep and cattle. But we will see that this is not the case for this gene in this case. Okay. Well, we see some other that are quite interesting. This is the cubilin that is the transporter of the cobalamin. So basically, I will tell you some more. So, but most of them, looking at the function, at the cellular function, are at a very basic level in the cellular function. And looking at the gene expression patterns, they are mostly expressed in many tissues. So it is quite challenging to, saying, okay, there is a selection there because of this phenotype. So there is still a big gap between the genotype and the phenotype. I give you a few examples, though, like this one. These are the map. Oh, there should be some red dots here that disappear. Okay. Anyway, I tell you, I think it's the MAGE versus IBM incompatibility. Okay, anyway, in this case, for instance, this is FAM184B. That is one of those genes that have a clear classification in terms of tissues expression, because it is mainly expression in muscle, is associated to temperature annual range. And there is a clear association between the two different genotypes. This is the, sorry, there should be a genotype also some here. The TT genotype is associated to a low temperature, annual temperature range, while the other genotype, that is GG, is associated to a higher. So it is more concentrated, the GG genotype on the Alps, for instance, on the mountains and in Northern Europe compared to this genotype. I'm sorry that didn't show up. I told you that sometimes there is a confusion between what we found and the traits that have been selected by humans, not by environment. In this case, for instance, this is a FST analysis of breeds classified on the basis of the corn shape or horn shape or horn and polled. There is a clear example, clear signal of this gene. So this is something that is confusing. I mean, it's most likely associated to horn shape than to adaptation to environment. Interesting, the ABCGT2, sorry, does not show up here for milk production. So it's not milk. So there's no confounding effect of milk. So looking at a few of them, this is the methionine sulfoxyl reductase. This is associated to oxidative stress. The reduction of oxidative stress has been identified with some interesting traits like calving easy in cattle and also gestational age at birth in humans. This was quite interesting for us, and the cubilin is the transporter of the intrinsic factor. The intrinsic factor is a factor that is secreted by the stomach and is very important to the transport of cobalamin. So the internalization, the absorption of cobalamin is really driven by the intrinsic factor, cannot be without the intrinsic factor. The cubilin is the transporter of the intrinsic factor cobalamin complex. Well, the problem is that we at the beginning thought, okay, this is linked to environments, to the food availability in terms of cobalamin availability. The problem is that the ruminants, I mean, cobalamin in plants is very, very poor, and in ruminants, basically, is the rumen microorganisms that synthesize the cobalamin. So we tried to see if the limiting factor was instead, as cobalamin is chelated to cobalt, if there was a map of cobalt in agricultural land, and indeed there is how the level of cobalt in the different areas is different, but so far we could not relate this to, I mean, we have to ask EPFL to try to see if they have a better map and to try to match this cobalt concentration with the presence of the different cobalamin receptors here. So what's next? Well, having these genes and also the other genes, what we did is we analyzed, using the sequences, we analyzed the effect of the different variants that were within the genes and in nearby regions. And then we calculated the LD between the marker that we found associated and these variants after classification of high effect variant, moderate effect using SNP-F. And well, what we found is, for instance, that for these two genes, this is to give you an example, this is the marker we used for detecting the effect. So it was in the chip, basically. The marker itself had an effect. So SNP-F told that this marker was intragenic and had a moderate emissense effect, so it was changing the amino acids. And also for the cubilin, this was the case. But in some cases, for instance, for the LAP3, there was no effect of the marker. So the marker we used was in an intron, but nearby there were a number of variants that were causing the protein to change amino acids. And some of them were quite linked, this is LD, to the SNP. Of course, this is to try to find the variant and to see if we can also identify the causal variant of the association we found. It is clear that in this case, we are not looking at or not expecting high effect variant like stop codon, because we are just looking at variants that are functional in different environments. They're not, the recessive genes or mutation. And then testing the effect of the variant on protein structure, we collaborated with Giovanni Chilemi. This is the cobilin, and this is the gastric intrinsic factor, and here is the cobalamin. The mutation we found is quite far away, so it's not in the place where the two proteins interact. But it still has to use molecular dynamics to see if a mutation in this amino acid is really changing also the structure in the interaction, in this active site or in the interaction point. And this, this is in humans. Have been found a number of mutations associated to different efficiency of the cobalamin to absorb the cobalamin in the gut. Well, I don't think I have time to describe this. This is very interesting because it controls meiosis in the proteasome, and you know that during the non-reproductive season, the males have a very poor sperm quality, and even they regress the testicles sometimes. So maybe another good gene. Okay, so the conclusion is that adaptation is very complex, and I think Henry agrees that probably involves all the genes in the genome. Okay. But some genes appear to have a relatively major role and are identified in several species using different techniques. So I think we should really concentrate on those. And one of these techniques is landscape genomics. It gives some hints about the variables responsible or associated to selection pressure. But the gene to phenotype gap is still large. Okay, and this is, we are working, the community is working to bridge this gap. Also, the identification of variants is challenging, and I think that genome sequences and also molecular dynamics, I like this approach in these days very much, is helping identifying or excluding candidate variants. In any case, I conclude with this statement that genome diversity remains the basis of adaptation, and conservation of this diversity is a priority to understand the biology and the breed for future needs. So with this, I thank you very much.
Thank you, Paolo, for this very interesting topic on landscape genomics, because also climate change can be connected to that. I went to a course with Elio in Ethiopia, which is extremely interesting. It's complicated, but the results are really giving us already a glimpse of what adaptation what you said indicated. So there's time for a short question.
Are you also thinking of including maybe in future in the model of landscape genomics also epigenetics, since also epigenetics partially represents the effect of the environment?
Oh yeah, this can be done, but I think before reaching epigenetics, I think we should probably work on including and controlling, the husbandry systems that are used in different breeds, to control also, to refine the climatic variables and accord them. For instance, why not using THI as instead of a single variable as the target to go for heat stress specifically? But this is going to happen in the future, I think.
On top of that, I can say that it's very important because we have a functional annotation of animal genomes where we want to understand, but each base in the genome will have an effect on whatever, which is also very important when you find something, when you can relate back to maybe adaptation or other traits. So I think that's
Consider that at least 50% of the QTLs or QTNs in humans are not in protein coding regions, but they are regulatory regions. So this is very important annotation.
Thank you, Bob.
Thank
very
you.
much. Thank you.
Okay, so now you have seen that we can have very funny stories or exciting stories about from gene bank samples. But in the next session, you will see more information on generic tools, which can be used whatever the target, the trait, or the environment you are interested in. So with the new SNP chip, multi-species SNP chip, that Richard will present to you. And then we have the data integration image portal that is under preparation and soon operational to help any of you find the data for your questions. For the moment, Richard, you have the floor.
Okay, thank you very much, Michelle. What I want to do is today is explain and tell you what we did in Work Package 4 and in developing a multi-species SNP array as being a new standard to compare gene bank collection for that. With this, we used information gathered what we did in the whole Work Package 4 by sequencing, by doing such landscape genomics where we took the genes and the variation which we put on these arrays to be sure that we will cover all these aspects, what we did in that. And of course the most important question is, okay, oh sorry, I have to, okay. The most important question is how to open these genetic reservoirs and what to store in the future. That's our main things which are of interest. And of course, you can put it in, but what do you put in and when do you put things in? That's a very important thing. And what is the difference between the different samples you will store? Of course, you have phenotypes, but of course, we also want to see, okay, what is the molecular base on that? So what are the molecular markers on that? So variation, it's all about variation. That's all what we saw in many of the presentations. Variation is the base of also doing selection or to select or to improve a variation. So you have phenotypic variation where you can show on the outside in many cases. And you have genomic variation, which you can type with molecular arrays on different platforms like the Illumina and the AFI platforms, which are available for many species. But how to capture this? Of course, it's phenotypic variation. You can monitor this, and you can, of course, you have species, you have breeds, you have animal idea, you have sex, you have relationships. What is stored, what you want to store, what is the age, tissue, blood, all this information which is part of Work Package, also Work Package 5, which we are putting into databases. That's the first start. And if all these centers will do that, then we have a very nice reservoir and overview of what is there and what is stored in all different countries of the world in principle. So that's the ideal situation, of course. And then of course you have molecular variation. And that is something where we want to either sequence and refine differences in the sequence, where we have an animal idea. And what would be nice if we could connect this animal idea to the information what is there in biosamples, and also connect this to that. And all the information which we get in the future, we also can connect to this database system, and that will help us to see what is there, and we can build on things and much more easily. I will focus now more on the molecular variation, and the whole point is, okay, there is variation which is captured in platforms, in Illumina platforms, so arrays, on AFI platforms, and there are many types. So going from 7K to 770K and all the different variants in there. There's so much variation, things are changing so fast that it's quite difficult, okay, what to use? What is the best thing to do? And is there one thing? In some cases it is. For example, I have to say, in the cattle world, I think it's the best example of that they really joined forces, because if you go back, or you go up from 7K to further, all the markers of most of the markers are also in the next, in the larger ones, or going back also 7K are present in, say, in the 43K, etc. But that's not in all cases. There are cases, for example, in chicken, where you have 60K and 660K, where only 1500 or 1400 markers are overlapping, which is, of course, is a tragedy when you use one system and one other system, and you want to compare things. So that's something, that's why what triggered us to start up in making a different approach. But first of all, we want to have and capture more variation, because most of those arrays which are there are biased to commercial populations. So what we did in Word Package 4 also is that we sequence traditional breeds, and an example of belted ones in cattle, chicken, and in pigs and in goats, as an example of variation of animals which we sequenced in more depth. So what did we do? We sequenced for a coverage of 10x, so 14 breeds, 60 cattle individuals, 121 breeds in chicken, over 300 individuals, 31 breeds in pigs, 100 samples, and 67 breeds in, with 100 in sheep. That will give us a pool of SNP information. So what we did on the sequencing, there was of course a lot available on the public domain, and what partners did put in as information, what they did by themselves. So from this pool, we have a very nice thing where we can select from. Of course, what is out there has to be aligned to the latest reference genome, because reference genome changes every time, we get more information, and of course we convert all this information to the latest reference genome for every species we worked with. But why would we make a multi-species SNP array? So of course there are several reasons for that. So one of the reasons I all indicated is that arrays are changing all the time. So when a commercial array is there, maybe the next day the commercial array is gone, or it's a completely different set of SNPs, so it's annoying, especially when you want to compare things. But of course, the most important thing is that we want to genotype populations, and you have in situ populations, but also you have the gene bank collections, which are important. And of course, there's overlap. So what is in both systems? And of course, what you can do is you can store material, update collections for that. Of course, what to select would be nice if we would, besides the animal itself, that we would have genotype information to also prove what is going on there and what you're going to save. But maybe you want to go back, so maybe you want to have information which is stored in the gene bank and introduce that back into the population because biodiversity is going down, or you want to go back in specific markers which are, say, lost due to selection in there. reintroduce, but also maybe you want to create new breeds. That might be also interesting to use the information for that. So what we came up is we got a multi-species SNP array, where we said, okay, we want to have 10,000 SNPs per species. The cost should be low, and low means that it will be below $20 per genotype, which includes the array and the genotyping platform. Not the DNA extraction, but I call that a separate thing, but at least, so including the genotyping at the provider's place. We aimed for at least 10 species, but we start with the major farm animal species. It has to be accessible worldwide, so that means that it should be wherever you are in the world you could use this. Public available, and there are no restrictions. So no restrictions means that you don't have to ask permission from somebody, from a company, to use this array. No, it's there, it can be used, and it would be nice to use it. Of course, in order to use it, you want to know what is on it. What kind of markers are there, which makes it interesting for you also to genotype. The first array which we made is the major farm animal species, so it's cattle, pig, chicken, goat, sheep, horse. The array is called IMAGE 001. We can imagine that we were going to make more. Yes, we do, because we're also going to work on IMAGE 002, which is close to finished. So this one is already finished. I will talk about a little bit more in detail with some of the preliminary results what we have for the genotyping. IMAGE 002, we would like to submit the SNPs for the development of the arrays half of February. So that's nice, and we're also planning to have a third one that will be outside the IMAGE, because the IMAGE project ends at March 1, but we still have the possibility to make, and then we focus there on fish species, because also there it's enormous interest of fish to be genotyped, but also for SNPs to be public available, not restricted by companies, because in many cases companies are close collaborating with researchers, and they sequence things and they get SNPs out of their data, but they want to keep it from themselves and not want to share it. So that's one of the things we try to avoid, and we are trying to use the information which is available. Not that we're going to sequence animals ourselves, but we're going to work together. That's the same what we did in all the three or the two other ones which we developed or going to develop, is that we work together with members, colleagues outside or inside and outside the IMAGE project to help to get this done. And I think there's a lot of interest to do that. Okay, the multi-species, what will we put on there? So 10K, what we will do is we have a common set of SNPs, and that will be around 80% of the array, which cover SNPs which are present in existing arrays, so that we can compare information which is already there. And of course, we will have populations included in there, so with minor allele frequencies. There will be inheritance panels, so you can use it for all kind of things to do that. We have ancestral SNPs in there, so that we can have a distinction between ancestral and derived alleles. And in some cases, and I will show some examples of how we did this for a few of the species we included. Traits, which is quite important. So in many cases, we focus on monogenetic traits, but also production, disease, and adaptation. And to connect for adaptation, that's the work, for example, with Paulo de Tel on the genes they discovered. We took out variations and put on the 10K for sheep in that cases. Diseases and also, especially diseases and the monogenetic ones, are coming from the OMIA websites or what we obtained from researchers in the field. The sex chromosomes, which is also very challenging, because most of the sex chromosomes, especially the Y and the W, are poorly covered.
but also with new information, we could add a lot of markers for both of the sex-typed, so the mammals and the birds, in this case chicken, to add more markers for the Y and for the W, for in many cases for all species there. We would also like the mitochondrial DNA, because a lot of studies have been done on the mitochondrial DNA in the past to have different haplotypes. So we would like to represent the different haplotypes in the species on the array for every, and also we want to do that, want to have it for every species. And further on, we also had some place for the MHC, and sometimes it's called differently in different species, to have some interest for the immune gene, and we took some genes which are connected to QTLs. And of course, if you go for some of the species, half of the genome is QTLs, but we selected some of them to at least to have some additional information on that. So this is so an overview what we are doing, or what we did for every species, and what we're going to do also for the other species, if the data is available, because that's also one for the large farm. So for the first one, it's quite easy. For the next arrays, it will be more complicated, more difficult to get all these things, but we try to be as broad as possible for that. Examples for the horse SNP selection. So we had variants related to the traits, which we collected from OMIA. Of course, there are coat color genes and some of the dwarfism. All these things which we, where there's a genotype there or an association, we put this SNP on. The Y chromosome marker, especially in the horse, was the Y chromosome is poorly annotated in horse. But with the help of Barbara Walker from the University of Vienna, where she published just recently a large number of Y chromosome markers, we worked together, and she selected over 100 SNPs on the Y chromosome, which we could add also for the horse, on the horse SNP selection. So the major histocompatibility complex markers, which we could find in ensemble. The mitochondrial DNA for to the different equine haplotypes. Ancestral SNPs, which are coming from the work of Ludovic Orlando. So he looked for SNPs which are, say, polymorphic in the ancestor, not polymorphic in the current population, are SNPs which are monomorphic in the ancestor and polymorphic in the current breeds, and also ones which were polymorphic in the ancestral ones and which are homozygous in the current breeds. So all this information is quite interesting to use this information. And then of course we have informative SNPs where we have minor allele frequency. In horse we had 34 breeds, and of course sometimes it's difficult to get information of allele frequencies of SNPs in population, because it's connected to associations or whatever, so they're difficult to share. But sometimes you also can circumvent that, and that's also what we did with Molly McHugh. So we sent her a list of SNPs. Okay, are those, because she was not able to tell if this SNP is informative in their populations, in their study, but she could say, okay, the ones you selected are very interesting to put on the array. So by that we will have information, but it's not always one-to-one and always completely clear, but at least we had the minor allele frequency in the breeds we did examine. Where are the informative horse SNPs coming from? 34 breeds from different countries of the world. And of course we have to annotate them on the genome. These are all the chromosomes where we'll see the coverage of the selected SNPs. And of course, and then we have a list of where they're coming from. So we have informative SNPs, which are used also on the developed arrays there, so to connect that to existing data sets. We have OMIA SNPs, so 44, the MSC 200, chromosome X close to 400, chromosome Y 99, the ancestral SNPs 322, and then we have SNPs in genes which are connected to QTL regions. A total of 10,321 SNPs which we have selected in horse. Another one is cattle, which is the other way, is that the base for the cattle array is the 7K, which is present, so it has a lot of information, but we added on things there which we think could be improved. Of course, there are new variants which are detected, so color genes which are not there. So there are mitochondrial DNA to make sure that all the mitochondrial haplotypes are covered. The ancestral ones are added, and let's work together with Dan Bradley in Ireland, where we also have the same thing of SNPs which are present in the aurochs and compare them to the current breeds. I'll come back to that later to have a slide on that. We have the X and Y chromosome. We have the informative SNP, where we have a lot of information on the minor allele frequency, MSC complex, and gene regions, which we, for candidate genes for there. The ancestral SNPs, I think it's quite interesting, where we compared the European sequenced aurochs, and then it's 4, with cattle breeds from Europe, and where we selected SNPs which are either polymorphic or monomorphic or polymorphic in one or the other two groups. And by that we can also look for ancestral and derived alleles for these. And of course, again, the alignment on the genome, you will see that because this is a very standardized array, because the base is on this almost 8,000 SNPs, so that the coverage is quite much more precisely over the genome. And also there you will see the numbers on that. There's a summary. So we have horse, pigs, chicken, cattle, sheep. These are the number of SNPs which are on there, so there's a little bit of variation in there. So this is what we aimed for, and this is what finally was get, because if you have a SNP, of course, it also has to be possible to make an assay out of this. So sometimes they fall off, so they are not suitable, so then you have to get replacements for that. That's nice, but also you want to see what is if it's working. So therefore we collected material, and so 1920 samples, and we did this in collaboration with World Package 2, where Cyprios and also Paul Witcher from the FAO did send out an inquiry. Okay, if there's an array, are you willing to genotype your samples with such an array? There was a response from over 100,000 samples, which are the centers who are willing to genotype. So that was also that's very nice. Further on, what I did is we selected breeds. So we asked the different centers, are you willing to send in samples which we can genotype as a test case? And I received from 240 different breeds for the 6 species, the 1920 samples. And I put on the array, so individual samples, of course, to get the genotype, trios to get the inheritance there for some of the, not for every species, but some of the species. Of course, repeats, so we had samples which are over different plates. And I also add on pools, where I combined, for example, a horse with a chicken and see if we can extract both genotypes for that. This is an overview of the samples and the number of breeds. So in horse we had 63 breeds, 517 samples, up to goat, here 27 breeds and 207 and everything in between, which is genotyped. In this case it's performed at Eurofins. And we just recently got the data, but also I can show some of the first glimpse of what the data. So we are still in full analysis of that. The number of markers, okay, what we're going to do is going to see what's the percent of markers which are typed, which is a little bit biased compared to what you can see on percentage. But we have one plate who was a failure, so we have to be done again of 284 samples. So probably also there the number of animals and the different species will go up. But if the marker was typable, the call rates are quite nice and quite high. Well, I said, okay, so this is the samples which are genotyped. There are some dropouts, but that's because of the one plate who failed. So as soon as we have this new data, I think we will fix this quite nicely, and then I think it's a very nice tool. What are we going to do with this? Because it's nice, but you want to compare it to something. So what we developed, and also in collaboration with Web Package 5, that we would have, say, a base PCA plot. So here we have genotypes from public available SNPs data set where we extracted the 10K, what was there in porcine. We extracted the SNPs, the information, and put it into a PCA. And where we have, say, the Asian breeds, the commercial Asian domesticated. This is the European domesticated, and this is the European wild animals, wild boars in this case, as a base. And this is one which you genotyped samples from different institutes, what we did get. And also here what you can see is that you get a nice separation of the Asian breeds in this case. And of course what you're going to do is you're going to put this on top of this also that you can show where my breed is coming from. So this is one of the examples which you're going to do. Another example, for example, the mitochondrial DNA. So we use this information and we clustered them, and we could find them that they're very clustering very nicely according to what we already know on the haplotypes for that. The Y chromosome markers, we are still working on this because there are SNPs, of course, which are segregating because they are present on the Y and the X chromosome. And also in some cases we know that is there. We have a lot of dropouts. Dropouts means that if you have a female in mammals, you don't have any genotype for this one, and then if you have a number of that, then the marker will fail. So we have to restore this information, and we work on that at the moment to get this whole data completed for that. And then, so it's there. You can use it, but there will be an update. And the update is that there will be an IMAGE 001 plus, I call it now for the time being. And that's because there are markers which are not working, not efficiently, so we can replace them. We can do that in one go for all different species. So we can delete the markers, we can replace them for new markers. So if there are still interesting markers which you want to add, please let us know. The plan is to do that within 3 to 6 months, so I have to see how things can evolve. And then, of course, meantime, we also work on a list of the whole marker set, where it's coming from, which trait, for example, and then it can be genotyped. There are different providers. These are the two main providers. We did now all the work with Eurofins. Also, the second one will be done with Eurofins. Eurofins is also here. Frederick is here if you have any questions on that. But in principle, it's free. So you can order it and you can genotype it with whatever provider it's available. These are the two major ones. We made appointments to them at least that they will charge you a fee of less than $20 for sure. So that's something what is important to do. And then of course there are recommendations. So what we say, okay, what is important there is that the samples which are stored in the Center of Genetic Resources, it's nice to collect genetic material like sperm and other things, but also collect blood for DNA extraction. Type the animals, genotype them with the preferable with, of course, with the IMAGE array so that we can compare. Use the tools which are developed also in WebPackage 4 and WebPackage 5. Samples should be submitted to biosamples, and genotypes can be connected to this biosamples idea to have a match with that and make it also public available. And by that I would like to stop. I would like to thank many people. So what we did is we assigned for every species let's say a species leader who helped us. So not everybody is on the slide, or there are missing people, I have to say. Most important, I would like to thank to Rainer Perez, who is in Wageningen, who combined all the things. He's working on the IMAGE 2 array and also on the 3, in connecting with Affymetrix to make sure that all the different information coming from all the different people are collected and shared in a good way. And of course, many thanks to Affy to make this all possible within this budget and time frame, because there was a lot of stress to get things done in the right time before the end of the project, and I think and it was also one of the deliverables that you at least have a multi-species array, and now we also can prove that it's there, it works, and by that I would like to stop.
Thank you, John. So thank you very much. So there is time for one or two questions because we started a bit. Yes, Ella?
Thank you very much. While it was obvious what species you would pick up for the image 001, I wonder what was the reason for selection of this species for image 002? Like for instance, why you have pigeons and you don't have turkeys or goose? Yeah. So what was the reason behind?
We looked at the survey, what is there in the different centers of resources, and so that's why we more or less compiled the choice of that. Also, the next thing, of course, it's important that there is preferable a genome, that there are SNPs, public SNPs available, where no companies are behind to restrict you. So there are all of things. So yes, maybe would have another choice, or maybe a better choice to have the follow-up. And for the other arrays, but sometimes you have to make choices, and that's the only explanation I can give.
It is multi-actor and multi-information.
Yeah, it's quite complex.
Yes?
Can you validate the chip on grids that are not in the discovery panel? In what is the...
Oh yeah, no, the validation is on data which is coming from the Center of Genetic Resources, which have not been genotyped yet. In some cases we have information where we have sequence information and or say 600, 700K information. We also can compare the results for that. But most of the information is from samples where we don't have any information from. So with samples which have to go into the bio samples archive, and where we can just add this information on top of that.
And what is the percentage of the SNPs that are not polymorphic?
I can't tell yet, because we just received it and we are full in analysis and making the other one, so that's, yeah. But we're working on that, and we will, of course, that will be all public. So we will share this. We will share all the information. Of course, we will write the manuscript out of this, but it will also be public available, the information, before we have the paper done.
Will you also check the imputation? Using 10K, imputing up to 50K?
We didn't do that, but it is possible in some cases. So it's possible, but we didn't look at that yet. Because we focus now more on the design and did it work and replace and what kind of SNPs should be on there. The next thing, of course, is that things are possible to do that.
For the imputation, there is a specific work ongoing for Chile because we have these two blocks of data with one chip and another chip and only 2,000 markers. So even without the technique, we are working on imputation for chickens and for cattle. It is rather routine, but for the other species, it depends on each consortium working on the species.
Yeah. So that's, yeah.
But for chicken, we try to propose something from image. Okay, so
and in the back. Oh.
It depends on how many SNPs will fail to be genotyped in the original in this data set. But is there any chance to replace the failed genotype, the SNPs that were failed to be genotyped by a panel of correct for ascertainment bias?
Yeah. Now what we do is we will so now we have So we have this. Now we know the genotypes. We know which markers didn't work, and we can replace them with markers that will work on this assay. And bias, yeah, of course, there's always a bias, because the original ones are very biased to commercial ones. Of course, we have a lot of SNPs which are overlapping with the current arrays, but still we looked at the allele frequency, which is in so commercial and in the say more traditional breeds present for that. And of course, MAGE is also in restoring and helping small breeds. So this is, I think it's a very important tool to use for the future.
Yeah, thank you, Richard. Thanks again. And so now we will move to the MAGE data portal. So Alessandra will present the complementary tool of this SNP approach, where you can find everything you want to know on gene bank collections on the data portal.
Make it small.
You go down.
It's difficult to can't see.
Will you go down?
No.
Make it full screen?
Yeah.
Full screen?
Yeah, now you can.
Okay.
Thank you, Michelle. And as we were finalizing the objectives of the whole project, and in particular for Work Package 5, already now many years ago, a few aspects were we found really important. And one was, of course, the overall goal of the project is enhancing the use of gene bank material in general, and to the purpose of traditional conservation and so on. But enhancing, and this was brought up again in the conversation yesterday, is also maybe highlighting like new use in current practice in breeding, for example, and in the practical aspects of the livestock production, other than merely conservation. It goes back to what people from the government agency were highlighting today, highlighting again the importance of preserving data. Second aspect was that since the late 90s, a lot of characterization, genomic characterization, phenotypic characterization projects were funded by EU and national agencies, and a lot of important and very well produced data were out there, in particularly for genomics, relative to this population that we are targeting. And third consideration, quite a bit of data work and databases and so it's been done again, like funded by this project, but a little bit scattered and needed some uniformity and some integration. Starting from that, one of the main goals of MAGE was the creation of this web portal that would integrate different data sources for material that was relevant to GenBank, but not only, beyond that, to this collection that we call genomic collection, but in general to collection characterizing the diversity of livestock in Europe. So the needs were for sure for integration from the various sources, an integration that has to be somehow standardized with consistency of all ontology to be able to make comparison, to be able to bring them together, to be able to be searchable in a very organized way. And again, we all know that with EU project, you build very nice databases, very nice tools that you put on the web, but later on, it's very difficult to maintain and keep updated and give assistance to users when the project is over. So we needed, to find something that was sustainable in time and was continuously updated and curated. And then, of course, you need visibility. Putting it out on the web is sometimes it's not sufficient to make someone, go to a broader audience as we were aiming to. And again, like we were collecting data from different gene banks, data from projects that were already deposited in different public databases, in private databases, many different formats. Therefore, we need assistance and help to the ease of uploading the information, to encourage people to upload the information. Still working on that. And of course, integration with other specific databases, and we're referring here to DADIS, EFABIS, as well as UGNA. So, building the conceptual model, main blocks of importance were rule set to ensure comparable data. We have to be strict with that. We have to be compatible with the archive that we've chosen, but also try to represent at the best the collection that we are collecting data for. Collect, standardize, and submit this data. Selection of archives based on data sustainability and persistence, as well as integrating the data with also support of novel technologies. And here, like I will repropose this picture a few times, is what we have as a general description, a simplified schematic data model for our MAGE portal. As you see, we have considered that data coming from gene banks were stored in a variety of different methods and formats. So from a good number of gene banks utilizing CryoWeb, which let's say consider the gold standard in this field, from gene banks with other very well organized databases, such as French, CRB, and other kind of different formats and collection, as well as data already, as I said, published in the other public databases. So we go back to this one, but we build an inject tool which is capable to collect data from the very many different formats, import data, validate them, and we will go into the validation process, and submit it to the archive that has been chosen. Oops, sorry. So, to implement this standardization, we have developed a set of a rule set to standardize the metadata from the very different records, which utilizes ontologies to ensure the consistency of supplied terms and synonyms, and assist also in the process of submission to guarantee the standardization. And of course, this process was developed together with all the partners, with a little bit feedback, and is an ongoing process, because of course, we had to set a strict set of mandatory data that are guarantee the minimum compliance to the rules of the archives, but as well, you know, getting suggestion and sometimes realizing that what we were asking was a little bit too much, and data were not available even for the minimum set that we. So there is a possibility of discussion and continued development of the rules and refinement of the rules. And here to mention also that all the scripts and the work that has been done within WordPage is publicly available also as far as the developing part. So metadata rules enforce data types. As you see, data types as far as text or different formats, and requirements, which are divided in mandatory, optional, and suggested. And we also kind of give explanation for the suggested and possibly, you know, trying to give guidelines also for future collection of the information by suggesting collection of new kind of data. And of course, it restricts the allowed values for each data, both for ensuring high quality and as well, of course, the comparability. And this, of course, refers both to sample and to organism. So, and what happened then? How do we use these sets of rules? As you submit the information, there is, first of all, a general preliminary check on the structure, the presence of duplicates, and so on. And then a match with the standards and the rule set that we have developed to ensure that the minimum mandatory fields have been provided, and as well to standardize language, terminology, ontology with the rules that you have seen before. What happened then? There is a kind of cross check with the different fields that are provided on the data, which provides back, since we know our data better than everybody, provides back information and the possibility to fix and to check and fix submission of your information on a summary for the samples for the single submission. So, and here, work? Yes. So here we're going to this phase of the general model, validation and gain metadata standards using the inject tools. At this point, the choice of the archive. Choice was, of course, easy for us because we were very fortunate to have EMBL, EBI among the partners, which represents, of course, like the European base for data storage and bioinformatics activities. Is Brexit free, although our colleagues are working in UK because it's a European institution with bases in Germany. So, and in particular, biosamples, you may have heard of it, is stores and supplies description and metadata about biological samples. Currently is hosting about 12 million sample data, and the rate of submission is more than 16,000 daily submissions. As you can see, like in the quite amazing growth of the data size in the last 2 years. What is important about biosample mainly is really that assigns universally recognized unique identifiers. So that identifier is something that we could use for a number of use and applications, because it links together the organism, the sample, and information that may be preserved in other archives, such as the variation archives, where you can store genomic information. And of course, it provides a sustainable and secure long-term storage well beyond the end of the project. And with, of course, curation in terms of assistance that is provided by EBI to the biosample archive. And of course, it's been built so that it fully supports the metadata standards that we have developed within MAGE. So now, we'll store our sample here. But of course, one of our main goals was integration. How do we integrate information that were already there, namely, about breeds, description, and specific breed standards available by DADIS FABIS, and other information on countries and gene banks connected to Eugenia, as well as the omics information that Richard just presented that have been produced within MAGE, and beyond the information about samples that are already there as far as genotype sequences and so on, and how do we present that to the world. So the construction of the common data pool, what we have defined as the common data pool, is bringing together all the information in one pool. How does it work? It performs the daily synchronization of a gene bank or gene bank metadata that were tagged as being from the MAGE project. During the process of inserting data into biosample, a tag is added to samples that are relevant to the MAGE project so that we can bring it back. It collates this metadata with geographic information where available, of course. MAGE produce omics information, like from the 10K multispecies chip and beyond that, and cross-reference to external breed and gene bank information from DADIS and Eugenia. And of course, X serves as a back end of the MAGE portal to present information to the world. The MAGE data portal, this is how it looks like. Write down the address that we have decided to give. And it's presented. This is the home page. As you see, it provided single access point to hold the image data with pretty powerful and I would say efficient and quick metadata filters. Gives the opportunity to download filtered results and to access additional information. Angular has been used to model it, and it seems like very efficient and modern. As you go, for example, on the organism search, you get detailed metadata description on each gene bank record, gene bank contact information for access. It does cross-reference with, of course, Eugenia and DADIS, and we'll see. And coordinates automatically generate with geographic views. So this is, something that may come to an end. And again, may help to advertise your collection outside in the world where someone, may look for a similar environment, for example, on what is available on a specific country, on a specific region and areas. Gives you a possibility to perform advanced search based on all the attributes, all the metadata fields. So it's searched across all the data meta field. It returns organisms, specimens, and data sets, again, downloadable. And a search result bring directly to individual detailed pages. Here an example is a link to DADIS. As you click on Normand, like on the supplied breed, this will pop up automatically. And here is an example to the link to Eugenia as you target like the gene bank country, in this case the Netherlands. It provides summary statistics, which is automatically generated, self-updating, and interactive. And for the organism, for the single specimen. And now, how do you contribute to MAGE? Of course, this was another major point of importance when we started the project, because if you want people to get data, you have to somehow aid in the submission and again, like assist in the submission itself. And there are very many different situations. So we try to consider all of them. Gene bank, single gene bank with data uploaded on CryoWeb, with maybe a first major submission, and later on additions, addition of genotype information, and again, samples that were already registered in biosample for some experiments or reasons, some specific subpopulation that needed to be matched with gene bank samples. So we can to envisage a different kind of scenarios. And as well as I said, genomic collection, they were there like because they're being genotyped in EU project or so. In doing so, we developed the inject tool. As for use of the gene inject tool, we have developed a few documents that works as guidelines, and of course we are also happy to do it in interactive way if you contact us directly, and also a video will on YouTube we will provide you with step-by-step help in the submission, but we're there as well to help. And this is a first example of a submission for creating a submission, the very first step, creating a submission for uploading your data, where you really choose what kind of data format you want to provide. We do provide a template, Excel template, to aid in the process of transforming your data to the requirements, as well as tools for uploading directly from CryoWeb. And here, like I've got an example of a complete submission, which update the biosample records directly through the submission to the inject tool. Now, you can use some of the tools that we have developed for new submission also to facilitate georeferencing of the sample. So by simply using, by clicking on the maps, you know, these for, again, as an incentive to upload GIS information, which, as we have seen in previous presentation, may become of really quite an importance, both for scientific purposes, as well as for dissemination and a little bit of publicity of your samples and your collections. And again, by downloading CVS format, we have developed procedure to aid in the submission of genotypes of collection that are already stored, or, you know, at the same time performing uploading of your gene bank data together with genomic information associated to relative to the samples. And here an example on how we facilitate also the downloading of genomic information for the other archive, which is European Variation Archive at EBI, which collects information, genomic information for, and is collated again within the MAGE portal. And this, for example, has fed information to what was developed recently as a diversity browser, as we want to also provide tools to analyze and give a simple representation of the variance that is present in subsets, contrasting subset of the population. And Jupyter Notebook was developed, which perform 5 different steps to provide analysis and interface this analysis in a simple way. How does this work? You have to create a reference data set, and once all the samples that have been genotyped with a 10K, for example, will be uploaded on the portal, you'll be able to create, by giving constraint, subset of the population that you want to use as a reference. And this is very much relative to the final objective, to the final aim of what you want to compare. It does locally perform the joining of the reference data set with the new data set in Plink. So the new data set is what you are comparing, and performs principal component analysis, as we have seen in the previous presentation, with statistical procedure and the orthogonal transformation, which just 2 examples can be used for, of course, exploratory data analysis, simple distance, characterization of the distance among the population, and for making predictive models. The important things that from a single entry point, you'll be able to enrich this information with phenotypic data that are collected in the image database to produce nice and visual representation of the population you are interested in contrasting. Here is, for example, by continent, and here is all the example with pigs with all the different breeds. So, as a summary, the Image Web Portal is now up and running. The inject tools is working and is ready for you to use it and aid you in the submission. Currently, we are hosting about 32,000 samples belonging to about more than 13,000 individuals in 13 species. The multi-species chip genotypes are being uploaded once all the checks are being performed. The diversity browser has been tested on a big data set, and we really encourage to use it on different subsets to get information on your population and collection. And this will bring us later to the next presentation. The portal is also feeding breeder interface, which is run through the MOPS software for simulation. So again, we facilitate the uploading of information from the image portal to MOPS to again enhance the use of a population with simulation that Simianer will be presented in the next presentation. And of course, again, here we encourage submission to the image portal, and any difficulties that you may encounter, please contact us. We're there and very happy to help. And most important, I would like to acknowledge people at EBI, which are really the basis of all the development of the validation tools of the common data pool and on the portal, and in particular Peter will also had the duties of keeping this Italian coordination of the work package a little bit straight and on track. So Peter Harrison, Alexei Sokonov, and June Fan, people at UCSC, Alder, myself, I didn't put picture because I'm here, Paolo Cozzi, with the development of the inject tool. People in Lausanne for the development of the geographical tools. Sylvain in particular, because it was giving feedback on the work that he's been doing with CRB and it was very important in the development of our portal, as well as a test, because he has submitted a lot of data, and people inviting him for the development of the diversity browser. Thank you. No, I kept it with no technicalities, but if you are interested in all, the back-end information are available, and they are publicly available also on the development on the scripts and all computers.
Maybe we will have to make
Yes, that's the thing. Like,
more precisely.
yes. And this was us further on in the after the 29. Exactly after February 29.
Do you all have to put in the information you have in the sample, or do you all, because sometimes there are restrictions on what you can add in? Is that possible, or do you always have to fill in everything? Can you elaborate
okay, there are mandatory data. There are, a minimum set, and that, of course, and then, of course, there can be addition, and what I should have mentioned, but I gave for granted, all the data are public. Okay, so this is, of course, didn't mention because it is of major importance to put your data out there and have people using it and to enhance the use. So all the data are public. What is going in is not coming out. But, of course, you can present availability of the information. So availability is there, but data that are going in are not going out. You can always have addition, but you have that minimum set. Later on, you want to add in, you add in because you have the reference information. Like the unique identifier will take you to addition to them.
Any questions? I think for the public data, what we have been discussing, and we probably will still continue discussing with gene banks, is sometimes it is a bit scary to publish information on existing samples, and still for me it's still difficult to understand why is it scary, because if it is on the web, the existence of the sample, it doesn't mean that the sample is free for everyone. It just lets people know the sample exists. Then each gene bank has rules for distribution, and there is very often confusion that if this existence of the sample is published, then it is available for everyone.
No, of course. I mean, there's availability. What we have found is that it's almost a philosophical matter of the property of the data, so the property of the sample, which goes back and is quite different in different countries and so on. But of course, there is always within the same, the submission for a single gene bank, there is always the choice of submitting some sample and some not. So again, you have the opportunity, even if the process is very much automated, to go in and click out what you don't want to submit. This has been done, for example, from the Netherlands, and that, relates to maybe the original submission of the sample to the gene bank and so on. So there is a choice there. But of course, what's not there is not shown. So.
If you will advocate to have all the samples into this system preferable of all the samples of genetic sources in principle of the world.
Well, yes, I think, once the system is there, another thing I didn't mention, but of course, EBI's archives are aligned with NCBI, so I mean, the idea could be to take it further, even beyond Europe, but of course, first, I'll be happy with the MAGE partner, okay?
Okay, thank you very much, Alessandra.
You're welcome, thank you.
Having all the genetic resources stored in the gene bank and used for comprehensive characterization, work package 6 now deals with using the genetic diversity in breeding programs to restore the genetic diversity in populations or to transfer certain genetic features into a contemporary population. And we will start with the first presentation given by Anna Simiano, which deals with a software package to simulate different breeding scenarios, which has the nice name BOX.
Yes, I'm not so sure if everybody loves that name. I had the impression that Michelle was not so positive about the name, but I think you got used to it. Yeah, and what I want to present, so it's a long session, and I try to keep you awake. And I think it will be less technical than the last talk. So modern breeding programs and also breed restoration or diversity restoration programs are complex by nature, and I just picked a few of them. You typically see, when you see a breeding program, you see boxes and arrows. We have boxes which are
cohorts of individuals or semen storage or whatever, and then you have arrows, and these arrows are usually gene flow, so that could be selection or aging or reproduction or something like that. And the basic idea, which we had probably like 15 years ago, was why isn't it possible to sort of simulate a breeding program by exactly using this concept by, you know, drawing boxes and arrows and then run the program? And that's actually what we finally came up with. So these boxes are called nodes now, and the arrows are called edges. And nodes present objects, which can be individuals, gene banks, cohorts, semen, embryos, single genes, whatever, which have certain properties, which of course need to be defined. So you have to say, is it a cohort containing females or males, or what age are these individuals? And edges represent processes that transform one parent node to a child node. So that's not necessarily meant in a sort of generational set. So a parent node a child node can also be the same individuals, but at a later age. And of course, these processes also need to be described. So if it's a reproduction, then you have to simulate a meiosis and all that stuff. And the key is just to define, first of all, a comprehensive ontology of nodes and edges. So what nodes and what edges are possible and how are they exactly defined that can be used or that are necessary to represent an animal breeding program. Actually, we also use it for plant breeding programs. They are not so different. And such a concept actually can also be seen as a basis for a standardized breeding program description language. So a very unified way of describing breeding programs, which I don't think is existing so far. So what are we interested in when we think about breeding programs? So we are interested about genetic progress, but also maybe of maintenance or restoration of genetic diversity. Also of economic efficiency or the risk, which is linked to the variability of the outcome. And we have many ways to control these things. So how many animals to use, which animals to genotype, which animals to phenotype, which mating schemes to use, which selection techniques, and much more. So all these need to be defined and sort of optimized if you are in a situation to design such a breeding program. So how to optimize such a thing? I think the most widely used approach is by using the experience of the breeder. So breeders say, okay, we did it like that, and maybe we use some more animals here and some less here, or we do this type of breeding value estimation, and then things work well. I'm sorry, but I still have a cold. For some components, like for genetic progress, you have formulas, but they are often limited to very special and simplified cases, and you don't have these formulas for everything. You can do simulation studies, and there are different types. There are cohort-based deterministic simulation studies, programs like zPlan+, stochastic individual-based simulations like, for example, AlphaSim. And our idea was to merge these two components and have a stochastic but cohort-based approach, which we called MOPS. Now, what does MOPS stand for? MOPS is an abbreviation. It stands for Modular Breeding Program Simulator. And it's easy to pronounce by Germans because this dog breed, the pug dog, the German word is MOPS, but without a B. So it was, for Germans, it's a very intuitive name. And it's actually in our package, which is uploaded in GitHub, which is programmed highly efficient. So it uses C and C++ in these big things like genotype matrices and so on. It's highly flexible. It supports a variety of different trait architectures. And it has a lot of pre-implemented functions, so you probably can use every type of breeding value estimation that is on the market. Of course, you can download it and you can use it. The only problem is it's like two R functions, and each one has like 200 different parameters. You just have to specify these 200 parameters, and then you're in business. And for all those who do not want to work through these 200 parameters, we also created a web-based graphical user interface, which is here under this address, and which is rather intuitive and user-friendly. So the overall design of the software is that you use this interface to describe your breeding program. This generates basically a parameter file. Then you have data which can come from, for example, from the database that was just described. And they go into this breeding program simulator. So actually, the parameter file defines these 200 parameters. And then the stochastic simulation is run. So thanks a lot. And finally, you get an evaluation and visualization of the results. But you also can export all results and do your own analysis if you like. And then that's in the future. We also want to develop an optimizer level, but that is work in progress. That's not existing yet. So we have 4 major steps that need to be done. First, you have to define the breeding setup. So do you have 1 population or 2? How are they named? What are the traits you're interested in? What is the genetic architecture of these traits? Can, for example, also include genetic maps and genotype data. You can describe which animals have which phenotypes. So, for example, males and females can have different phenotypes, with selection indices to use. Then the next step is you draw your breeding program. And actually drawing the breeding program means drawing the breeding program. So this is from this FAO, prior conservation guideline. This is a breeding program for the reconstitution of a population from a gene bank. So that's how it looks like, and that's how it looks like in MOPS. So you basically draw the same thing with drag and drop, basically. Here you see in bracket numbers. So these are the number of individuals that are given, but this is a variable. You can change that. So the red ones are the females, the blue ones are the males, and so on. This is a simple one. This is a slightly more complicated one, but you can bring this to every type of complexity you wish. Then once you have drawn the breeding program, you push a button and say simulate, and then it simulates. And then you can analyze the results and plot the variables you're interested in. And for this simulation, for a single MOPS run, of course, you can replicate these things. So for example, this goes through in like a few seconds or so, the simulation. So here, for example, you have for the different cohorts, for these backcross generations, the average relationship within the cohort, and this is the average inbreeding within that cohort, which is quite high for the intercross. Here you have a phenotypic mean, so I have assumed these are sheep, and this is a daily gain, and that the recipient population is 20 grams per day above this gene bank. So you see with we have a decrease in performance with this backcrossing. And you also can look at the genotype proportions. This is just one sample or one SNP, which has a frequency of 0 in the recipients in the gene bank 1, and then you see the backcross 1, backcross 2, and the intercross, it's going up. So this is all implemented, and you just click and you get those results. So, of course, it can do much more than shown here. So you can read in complex genotype data and maps in all possible formats. You can create all types of genetic trait architecture, so polygenic or QTL, so mixed inheritance. You can have load in recombination maps. You can do that for single or multiple traits. We have implemented most cutting-edge breeding value estimation methods like BLUB, GBLUB, all the base methods, single step, as you like it. Of course, things don't get much faster if you use a very complicated breeding value estimation for a large data set, so that you should be aware. Of course, you can also simulate things like gene editing, and there's an expert mode which gives you much more possibilities, and there's also the possibility to add new features upon request. So if you have a specific thing you want to try and you let us know, you probably can implement it quite rapidly. And we have actually a follow-up project where funding is already secured, where the focus is to develop such an optimization tool which sort of allocates resources in a way to optimize your target function. It has been used in several demonstration projects, and we will have, I think in the next two talks, you will see some applications. We had a training course in November in Paris where we taught the use of MOPS to quite a number of students. You can all play around with it. We have a guest login, so you just can log in without a keyword, and you can play around with it. Actually, if all of you play around with it at the same time, our server may crash, but you're invited to do so. If you're interested to use MOPS in a serious project, let us know and you get a personal account, and I think we also probably should talk a little bit about how big your project should be. And also if you need additional functionalities, let us know. So that was it. Actually, the main developer of the code, especially of the R package, is Thorsten Pog, who is here. He cannot be here today. Because he's since 1st of February funded from a different project. But this project is actually this project where the optimization stuff is going to be produced. So that's it.
Maybe I have one. How can you integrate genome editing or gene editing into this program as a single trait, or how do you handle that?
Yeah, you just can say, okay, I have one of these arrows is replace this SNP by this other SNP.
So the program simulates a breeding program. Will it also be possible to optimize or simulate the complementarity, conservation, and breeding program in situ, ex situ?
Absolutely, yeah. That's, yeah. Yes, the answer is yes.
Yeah, I think
when you do mating, can you also design not only just make the best of best, but for different types of mating?
Yeah, we have optimum genetic contribution implemented, and you certainly also can sort of order animals according to breeding values and then mate best with the best or something like that, yeah. You can actually, I think if you really want to do a big thing, then actually it would be our recommendation to install the R package on your server and then send this JSON file to your R package, because it will not eat up our resources. Yeah, it's possible.
Good. Any other questions? Thanks again. And now we follow with case studies. For the first one, we go a little bit further away to Colombia, to South America. But presentation will be given by Gabor Pecharo, and talks about the breeding program of the Creole cattle in South America.
Okay, so thank you very much. This presentation is about our task in the IMAGE program, which was actually a collaboration between BOCU, so the University of Natural Resources and Life Sciences in Vienna, Agrosavia in Colombia, which is a research institute, also INRA Guadeloupe, Wageningen University and Research, and Göttingen University, where you will see parts of the, where basically we were integrating with our Colombian and Guadeloupe colleagues the outputs which are coming from the MOPS, which was presented just a minute ago, and then also things with the optimal contribution selection by our Wageningen colleagues that will actually follow up, or more details on this will be follow up after this presentation, so stay tuned. Also just to give our best acknowledgements for the breeders and data providers for Colombia and Guadeloupe, and also, of course, to the European Union to funding the IMAGE program, which we are very happy about, and then where basically this made all this work possible. A small introduction on the Creole cattle. So it's initially introduced to the Caribbean in the 15th, 16th century with a multi-purpose meat, milk, work, and skin kind of use. And then, basically, a bit unsurprisingly coming from the Andalusian cattle, basically on the, from the European side. But then there were various integration events and admixture events in course of the centuries from West Africa, and also then Indian Zebu cattle from in the 19th century. So basically, then this Creole cattle was spreading throughout the Caribbean and also to the South America. So this is then what we call now, or we call under the umbrella term for our purpose, Creole cattle. Now, this also has, or the Creole cattle has also breed characteristics, and you see that there are some of these breeds are looking very different. So, well, maybe it's calling them all Creole cattle is a bit simplistic, but at least it's certainly easiest to put up a name for the presentation such as this. So anyway, so we have the Blanco Ojinegro, the Costeño con Cuernos, Romo Simoano, and San Martinero. These are from Colombia. And then the Guadeloupe Creole cattle is a certain different branch. And then just little arrows just so we can keep, place these breeds also on the map. So, well, the objective of our work in the IMAGE project, or in this task, was to enhance the breeding programs of Creole cattle populations in the mentioned countries, and using first the simulation procedures for MOPS, and then also after that see what can we do with the optimal contribution selection approach. So we did exactly that. So simulating a breeding program, that was our first step. So what we tried is implement a little bit with the country-specific characteristics, and also after that optimizing the breeding program to keep relatively high breeding values, or the production high, and minimize the inbreeding levels. So for the ease of basically presentation, we concentrate right now on the Colombian Blanco Negro cattle, or bone is just a shortened version. And then the trait that we will be using as an example is weaning weight, sorry, of course. So this is then our workflow. So the two programs that we were used are MOPS and the GeneCon software for optimal contributions. And also we are having genotypes, or well, including genotypes of this mid-density of 50K genotypes from these animals, or well, in this Banco Negro cattle. So we started with a breeding program simulation, and we started in a very, let's say, very general way. So what you could see here on the left or your right side is actually an extremely simplistic breeding program where we would have a. Is it just me, or I'm mixing? Okay, so we have bulls and cows, and then we are mating them together, having basically some offspring, and then so we do this several times. So there we did 10 iterations in this small example, and we have some selected bulls that we are looking at how they are doing in terms of production levels and inbreeding levels. So unsurprisingly, if you use such a scheme, then what we have is an increase in production levels, but also increase in inbreeding. So that is, let's say, just was a small case that we started up with. But then, of course, we wanted to do something that actually has a direct correction to the IMAGE project, and that is using gene bank material. And then our question was, how can we, or what happens if we use a gene bank material? So how it actually these parameters are changing, in particular the inbreeding level. So we did just that, and so we put up a slightly more complex breeding program, or well, this was with great help from Thorsten. So anyway, so if you look at, so it might seem complicated, but it's not as much. So basically, what we are having is a starting population, and then what we divide them into two. So basically, this part is similar to the one that we had before. And also this part is also, I mean, to have two unrelated populations. And one we call a conservation population, and the other we call a breeding population. And the conservation population is mostly for the, well, just maintaining and keeping high diversity. And in the breeding population, there are the animals that were, I mean, selected normally for, I mean, for high breeding values. And then, what we were looking at is are the diversity parameters. So in this graph, so the x-axis are the generations. So these are the numbers of simulations of generation, and the y-axis and the share of heterozygosity. So if the heterozygosity goes down, that means that the inbreeding goes up, which is not really beneficial from the diversity point of view. You see that so this diversity or lessening of the diversity or lessening of the share of the heterozygous markers is much more prevalent, of course, in the breeding line, or that one which was selected for actually the breeding values. In the conservation line is relatively stable. And now actually with this step we wanted to demonstrate what happens if we take animals and basically simulate the scenario when we take animals from the conservation line, that is the gene bank material, and then put it to our, I mean, just made it to the breeding program animals. And then what we are actually simulated or proven, that the inbreeding level is basically, well, the situation with the inbreeding improves somewhat, and basically that it's a beneficial benefit from the diversity point of view. It's beneficial to point. I mean, to do from diversity point of view, is beneficial to do these moves. Now, you also might say, okay, well, this is relatively obvious. It is, but also how we see also the benefit of all this, and especially the MOS program from the gene bank related kind of developments. The thing is, all the animals in the gene banks are genotyped. So basically, what you can use and simulate future scenarios without actually taking out any samples from the gene bank. So you have the gene bank genotypes, you have your breeding population genotype, you all throw them all in the program and simulate what would happen if I take out the semen or whatever for these bulls. And then if you like the thing, what happens, then you do that. And if not, then probably you look for some other breeding bulls or other gene bank bulls that you would take out to your real population, and then so that would result into a scenario that you like better. So basically, this is something that you can test. What would happen if I take out some sample of bulls from the gene bank? So I think it's a pretty neat kind of approach that you can use. Okay, so this was the first part on the simulation, but then well, we actually had animals and also, yeah, frankly, our goal is also not just to increase diversity, but also to increase production. And then that is especially the thing that is done by the optimal contribution selection. So this is the courtesy of our Wageningen colleagues, Harman and Jack, were the main people in this part. So in the previous part, so we had some graphs on the productivity, but basically that was a simulation. But we wanted to actually make an assessment what happens really with the materials or the on the genetic merit of the animals when we are taking into account the improvement of the production, but also the kind of having the diversity in a specific level of specific check. So here what we were using also was a trait of the weaning weight, particularly for this example, for this presentation. And this is more or less a summary graph that is showing on the y-axis similarity level. So this is the less similar the animals are, they are more diverse, and the y-axis is the mean estimated breeding value for weaning weight. And then, so this summary graph, or basically these results, also show that, and in this case, the current population is meaning that the breeding population, or the one that is without any intervention, so we are doing just plainly breeding or plainly kind of improving the production levels. And then in blue, then there is the current population, and then the cryobank materials. And then, so basically what we are seeing is that we basically arrive into a specified estimated breeding value levels, but also that we are better off with the diversity parameters. So to summarize that, so the using of breeding, sorry, so the gene bank bulls had a clear added value to the population. Well, we saw steady increase in breeding values, but also we could contain the inbreeding level per generation, which was a very well, good news. So, and to summarize all our work in the MAGE program, so that we did a study that discusses the implementation of selection in in this Banco Negro population of Carayola cattle in Colombia. So in the simulations, we show that the inbreeding levels could be improved by using bulls from gene banks. And with optimal contribution selection, we provide a sustainable breeding alternative for genetic resources. Thank you very much for your attention.
Thank you very much, Gabo. We have time for one or two questions from the audience. Any questions for Gabo? Everything seems to be clear. Okay, and thanks again.
Thank you.
Continue also with the optimal contribution with another case study that will be presented by Harman and Associates. Work of Harman and everybody from the Netherlands.
Is this one working? I'll use this one. Okay, so good afternoon, everyone. My name is Harman Dukas. I'm a PhD candidate in university and research. And I would like to continue with these case studies of looking at genetic diversity in cattle using optimal contribution selection, focusing on this trade-off between, on the one hand, improving genetic merit, and on the other hand, maintaining genetic diversity. Because this is a trade-off that we're commonly confronted with within breeding programs. So if we talk about dairy cattle, in my case, we want to improve genetic merit, for example, for either total merit, so the economic profit, or for perhaps specific traits such as yield traits, or perhaps other health or fertility traits. At the same time, we want to maintain genetic diversity, because by maintaining diversity, we can ensure that also on the longer term, we can sustainably select. And our question was, what is now actually the added value of gene bank pools with respect to this trade-off? And this was also one of the main objectives of IMAGE, is to show what is now the added value of using gene bank material in current populations. So we did this using a case study in Dutch Holstein Friesian dairy cattle. And this breed dominates globally the dairy cattle, the dairy production. And within the breed, high genetic progress has been realized over the last few decades, while at the same time genetic diversity has been lost. Well, since the 1990s, genetic material of this breed has been stored in the Dutch gene bank, and currently there are around 5,500 bulls stored, with approximately 25 doses of semen per bull. So as I said, the aim was to determine the added value of these old gene bank bulls in terms of genetic merit and genetic diversity. So we defined two groups of animals. On the right you see what we call the current bulls. So those were defined as the bulls that were born between 2010 and 2015. And the gene bank bulls, which were bulls from the gene bank collection from before that time, for which we also had genotype data available. And what we then did is we compared two populations, one in which we would only use the current bulls and one in which we would use the current bulls as well as the gene bank bulls. And then, as I mentioned, we're going to try to improve genetic merit while at the same time maintaining diversity. So for that we need measures of both merit and diversity. So for merit, we use estimated breeding values for each of these four different indices. So the first one was the total merit index. The second one was a yield index, so that included milk, fat, protein, and lactose yields. The third one was other health, and the fourth one was fertility. Then as a measure of genetic diversity, we used the expected heterozygosity in such a population. And then we used, as was already explained by Gabor, we used optimal contribution selection to achieve the maximum amount of genetic merit at a certain level of heterozygosity. So the figures that I will show you are very similar to the one you just saw in the presentation of Gabor, where here on the x-axis we actually have the diversity. So in this case on the right part, we have the highest diversity, the highest heterozygosity. And on the y-axis, we have the merit. And then we look at the population before we do any optimization. So that's the box in this case in the middle. And then our target is to go to the right upper corner, because that would mean a high diversity while also having a high merit. So now the results. First for total merit, and before we do any optimization. So if we look only at the current bulls, so the ones born between 2010 and 2015, then we see that on average the heterozygosity is around 34.5%, while the total merit is around 140 or so. Well, if we look at the population where we also include the gene bank bulls, then we see that we move more towards the right, so we gain a little bit of diversity, so a bit of heterozygosity. But you also see that the merit is quite lower, because these older animals generally have lower breeding values. Now then the first thing we can do is, when optimizing, is we're going to maximize the diversity. So we're going to try to go to the upper, or the most right position in this figure as we can get. And then you see that with the gene bank bulls, we can get to a heterozygosity of 36.2% compared to 35.5% if we use only the current material. And in both cases, you see that maximizing diversity comes at the cost of merit. So we go down in merit. Now we'll add the curves where you actually look at all these trade-offs between merit and diversity. And this is the curve that you can get when using only the current bulls. And this is what you can get if you also include the gene bank bulls. Now what you can see here is, these are the numbers of animals that are selected by optimal contribution selection. So you see that if you focus more on improving genetic merit, then it will only select 10 individuals. Well, if you focus more on diversity, then it will select a lot more individuals. And what we can see here is the added value of gene bank bulls is the difference between the two curves. And if we look at the left part of the graph, so if we focus more on improving merit, then you see that these curves are approximately the same. So there's no added value of having these gene bank bulls in there as selection candidates. Whereas if we move more towards, we focus more on diversity, you see that there's a bit of difference between these curves. So there is some added value. That is, we can get a bit more genetic merit if we use these gene bank bulls at the same level of diversity. Now this is for the total merit index, and we did the same for the sub-indices. So first for yield rates, and what you then see is that also at lower levels of diversity, there's still some added value of having these gene bank bulls in there. And for higher diversity, this difference is bigger again. Well, then we did the same for other health, and the same for fertility, and especially for fertility, you can see that the difference is quite big, which is because there are quite some old gene bank bulls with high fertility breeding values. Now to visualize this a little bit in a little bit different way, what you can see here is the scenario in which we use all the individuals as selection candidates, and then we look at how much of the total contribution should go to either one of the two groups. And here again on the x-axis you have diversity, and on the y-axis you now have the contribution. And what we see for the total merit is at the moment that we do not focus on diversity, so we focus mostly on merit, that we only select current bulls. However, if we focus mostly on diversity, then 80 to 90% of the contributions actually go to this old material. Now we can look at the same figures for the other traits, and what's then especially noticeable is that for fertility, that wherever you are in terms of diversity, we always select 80 to 90% goes to the gene bank bulls. Now this was for the Holstein Friesian breed, which is a large commercial breed, and we also did this for a local breed in the Netherlands, the MRI, which is after the Holstein actually the largest breed in the Netherlands. And for this breed we had less information available, so we divided the information in two groups. We had around 120 bulls born after 2000 and almost 300 before 2000, and we optimized only for the total merit index. And the results were actually fairly similar to the ones for the Holstein Friesian. So here you see the scenario for maximizing the diversity. You see that in both cases you can move quite a bit to the right in the figure, and we can get a lot further when we use the older bulls as well compared to using only the current ones. Yeah, so these are the values. And then we also had two scenarios with a bit more focus on merit, and then you see that the difference between the two populations is quite a bit smaller, but there is some difference, so there is some added value of using also these old gene bank bulls as selection candidates. So in conclusion, I would state that gene bank bulls can be effectively used in both commercial and local breeds to increase genetic diversity. When we consider the trade-off with genetic merit, then it seems that there's limited value for the current total merit index. However, if we look at specific sub-indices, then this value becomes a lot larger. And this is important, especially because breeding goals, they change over time. So what is currently the total merit index will not be the same total merit index in 20 years or so. So the components will get different weights, and perhaps we will also include new traits. And for those new traits, there might actually be valuable information in the gene bank that we're currently not aware of. So from this work, there were two publications, as you can see here. If you want to know more, you can read those. And thank you for your attention, and I'm happy to take any questions.
Open for questions.
Thank you very much. Are we going to do what you observed in your simulation?
Yeah, well, I will present my work this end of this month or beginning of March at CRV, so we'll also include this part. They are still in the process of moving towards controlling genomic relationships rather than pedigree-based. But I think, as you can see, for the current total merit index, there's not a lot of added value. So they are currently more in the left side, actually, of these figures. So in that case, I'd say, at the moment, probably no, but perhaps in the future.
Any other questions? Okay, then thanks again. For the last presentation, now we move from cattle to chickens, and the presentation will be given by Claudia Dirks, and you will learn about how to paint white-shelled eggs blue.
Thank you, Stefan, for introduction. My name is Claudia Dirks, and I'm from the Friedrich Löffler Institute of Farm Animal Genetics in Mariensee in Germany. And today I'm going to tell you a story about painting eggs in blue. This is a joint project in MAGE between our institute and the Lohmann Tierzucht GmbH and the University of Göttingen. The objective of our case study is to give an example how to use a specific trait of a genetic resource population conserved in a gene bank by transferring it into a commercial breeding line. In our model for marker-assisted introgression, we take the blue eggshell color of Araucana and introduce it into a high-performing white leghorn line, aiming at the final population, a high-performing blue layer line. This high-performing blue layer line will be homozygous for the introgressed blue eggshell color locus, has a maximum of similarity to the white layer line, especially in the proximity of the target locus, and shows a high genetic diversity. Is there any commercial viability for blue eggs in Europe? I don't know, but in Great Britain or in the USA, blue eggs are requested products. First, I want to explain what introgression is. introgression is the introduction of a trait from a donor population into a recipient population. The trait will be fixed in the recipient and the donor genome should be minimized. Introgression is a tool for genetic improvement. Genetic resources may hide genes for agriculturally important traits. Another aspect for introgression is to map genes or to analyze their function by isolating and transferring a gene or a chromosomal segment in a different genetic background. This may help to dissect the genetic architecture of quantitative traits. This is a basic breeding scheme for marker-assisted introgression. You can see that we take a trait of interest or a gene of interest. This is this red dot, and introduce this red dot into the recipient. The F1 generation will be back-crossed with the recipient, and so on. On the one hand, we have to do foreground selection. This means that we select on each back-crossing cycle only heterozygous individuals, and at the end we only select the homozygous individuals. And on the other hand, we have to do background selection. This means that we select on each cycle of back-crossing against the donor genome. Between our generation, we observe recombination events. This means that there is a crossing over between both DNA strands, and we get chromosomes that are mixed of the donor genome and the blue is the donor genome and the recipient genome. To do this introgression very efficiently, we use markers that are distributed over the whole genome, and some alleles of these markers are specific for the donor and other alleles are specific for the recipient. Now we have a look on the trait of interest. Why are chicken eggs of different colors? The normal eggshell consists of a white calcium carbonate matrix. In brown eggs, the pigment protoporphyrin 9 is stored in the cuticula of the eggshell, and in green or blue eggs, the bile pigments biliverdin and biliverdin zinc chelate are stored in the eggshell and in the cuticula of the eggshell. The genetic background of blue eggs is very simple. The blue eggshell color is inherited in a dominant way, and the causal mutation is a large retroviral insertion of 4.2 kilobases in size, near to the gene SLC01B3 at 65.17 megabases on chromosome 1. The gene SLC01B3 encodes a biliverdin transporter. Then this insertion leads to an overexpression of SLC01B3 in the oviduct. The biliverdin concentration in the oviduct increases and is being stored in the eggshell. A dosage effect was observed for this mutation, and we see that on the left, the eggs of homozygous hens are more intensely colored than the eggs of heterozygous hens on the right. This is the breeding scheme of our case study. We start with an initial F1 generation, followed by 2 back-crossing generation and final intercross generation, aiming at a final animal, a high-performing blue layer, white leghorn-like line, which is homozygous for the blue eggshell color. To be able to do selection, all animals in our case study were genotyped for their sex and the blue egg genotype by newly developed CASP, this means competitive allele-specific PCR assays. And furthermore, all animals were genotyped for a customized 52K SNP array and 24 additional SNPs that were developed at introgression locus based on sequencing and genotyping data of the founder. These additional SNPs allow us to detect recombinant animals, based on distinct breed line specific haplotypes for Araucana or White Leghorn. Between the generations, analysis was done, and we selected the best animals for the next mating. Normally, we would expect a White Leghorn genome content of 87.5% for a BC2 generation without selection. Our selection criteria are that we want White Leghorn haplotypes at the introgression locus at first. Then we want a high similarity to the recipient White Leghorn line genome-wide, and also we want a high genetic diversity. Recombinants were detected by Merlin, and other selection steps were done using the programs MaxSafe and MOBS. For the mating of the BC2, we only selected the cocks out of the recombinant BC1 cocks. Therefore, all BC2 animals in our case study were recombinant at the introgression locus. This is an analysis of the chicken of the backcrossing generation 2. We estimated the mean percent of White Leghorn genome-wide based on founder-specific SNPs. And you can see the box plot for this analysis here. The red line marks the expected percent of White Leghorn genome for a BC2 generation. And we can see that the White Leghorn genome content was higher in our BC2 generation than expected without selection. It was 91.92%. And doing marker-assisted introgression, the gain of the recipient line was 4.42% in our backcrossing generation 2. This case study has not been finished yet. For the mating of the intercross, 20 cocks and 100 hens were selected for mating, and 1 cock was mated with 5 hens each. All of these animals were selected out of the heterozygous carrier based on the similarity and the diversity of simulated progeny of these animals. This simulation study was done with MOBS. And then 751 chicken hatched in November last year. The sex and the blue egg genotyping has been finished to date. And we observed 188 homozygous animals for the blue eggshell color genotype. 367 animals were heterozygous and 196 were homozygous for the wild type allele. As expected, we observed Mendelian inheritance for the blue eggshell color genotype. These 188 homozygous chicken will be used as a basis for a new high-performing blue layer line. Currently, the SNP array genotyping for the intercross chicken is underway. In order to analyze the success of this introgression, we do performance tests. For the F1, or the F1 was kept in floor pens, and the data was recorded per group of only 37 animals and without a control group. And for the 2 backcrossing generations, the hens were kept in individual cages, and we had the same number of controls in these both generations that were kept under the same conditions. The data was recorded from week of life 20 to 79. The BC2 is still in progress, and the intercross unfortunately didn't start laying. Traits analyzed were mortality, total egg number and laying rate, and egg quality traits, egg weight, eggshell strength, and eggshell color. These are the results for the laying rate. You can see, or the laying rate is divided into productivity intervals. One productivity interval is of 4 weeks, and interval 1 starts in week of life 20. You can see here on the x-axis the productivity intervals, and on the y-axis the laying rate in percent. On the top you see the results for the backcrossing generation 1, and at the bottom you see the results for the backcrossing generation 2. The blue lines are of the both genotypes for the blue eggshell color, and the black lines are the respective control groups. We can see that the laying rate is above of 90% in our crossed animals. This is much high. And unfortunately, we had a problem with the backcrossing generation 2 because the laying rate of all groups of the BC2 decreased since a week of life 45. And it seems that the white leghorn group was better to compensate the stress. It was maybe because of high temperature in summer that we observed this decrease. But we think that this decrease is not representative in general. So in order to compare the different generations, I only concentrated on the productivity intervals 3 to 6. These intervals include the weeks of life 29 to 44, very high productivity intervals, and we see that we observe for the BC1 a mean laying rate in this interval of 92.3%. In the BC2, a higher laying rate of 94.7%. This is about 2.5% higher than the BC1, but also 2% lower than the laying rate of the white leghorn. But we think that the laying rate increases with a higher white leghorn genome content. We also see that the non-carrier in our study have a slightly higher laying performance. These are the dotted lines. This is another graphic for the laying performance. You can see here the total egg number for the whole laying period. And on the left, you see the data for the BC1, and on the right, you see the data for the BC2. On the x-axis, there is the generation and both genotypes. BW, these are the carrier, and these are the non-carrier. And in gray, these are the respective control groups. For the BC1, we had data until week of life 79, and for the BC2, we had data until week of life 71. This is the reason why these both analyses are not in one graphic. We analyzed the data with the Tukey HSD test, and groups which share the same letter are not significant from each other. And we see that the BC2 non-carrier are not significant different from the white leghorn line. What we see else is that the BC1 laid about 20 eggs less than the white leghorn control line, and the BC2 laid only about 5 eggs less than the WL control line. Furthermore, we also see here that the non-carrier have a very slightly higher laying rate than the carrier. And the performance test or the performance data of the intercross generation will be quite interesting regarding if there is a possible influence of the genotype on the laying rate. These are the results for the egg weight. On the y-axis, the generation and the respective genotypes, and on the y-axis, the mean egg weight of week of life 40 and 49 in gram. It starts at 50 gram, and we see that the eggs of the F1 generation is significantly smaller than all other groups, and the important message here is that all eggs of the back-crossing generations reached the size MOL and weighed above of 60 gram. These are the results for the mean eggshell strange. It starts at 40 Newton. And we see that all generations in our case study have a lower breaking strange than the white Leghorn control groups. But we also see that the breaking strange of the BC2 animals is much higher than of the BC1 animals. So we can say that the eggshell strange is improved in the BC2, maybe due to a higher white Leghorn genome content. The next quality trait of the eggs is the egg color. These are pictures of our heterozygous hens. You can see that we observed blue to green eggs, and the non-carrier eggs were from white to a touch of cream, which you can see here. And in the middle you see the white Leghorn, and on the right top of this slide you see eggs of homozygous Araucana, which are more intensely colored. The eggshell is described by the LAB, the LAB values. These are 3 values. The L value is the value for the lightness. The A value is the value for the colors green or red. If this value is negative, the color is green. And the B value is the value for the color yellow or blue. And you can see here, I reduced it a little bit for better understanding. On the x-axis, you see the generation. I included the 2 back-crossing generations. I also included the Araucana, but I have to say that we only had a few eggs of homozygous Araucana, so these results are not really good comparable. But what we see is that the A value is influenced by the genotype of the hens. And in our case study, we also observed that the lightness increased from generation to generation. You see this on the top. And we also observed this touch of cream color in our population. And we observed that this touch of cream color decreases from generation to generation. So the green value is influenced by the genotype, and more white Leghorn genome content leads to an increased lightness and decreased yellow color. In summary, we can say that the marker-assisted introgression in our case study leads to an increased white Leghorn genome in the back-crossing generation 2, especially at the target locus. And we observed a high laying performance of the introgressed hens. The eggs were of good size, but decreased in their breaking strange compared with white Leghorn. And it seems that other loci influenced the cream color of the eggshell, but probably independent from the blue eggshell color locus. And we got 188 homozygous intercrossed chicken, and the genotyping is currently in progress. These homozygous animals will be founder, as I said, of a white Leghorn like a blue layer line, which is homozygous for blue eggshell color. The selection efficiency will be evaluated at the end of the project when we have all data by comparison of the simulated data and the real data. And we have to wait for genotype and performance data of the intercross generation, because this data will be necessary to make a statement about the introgression success. You see that the work for this case study is still in progress, and now it's too early to evaluate the efficiency. And at the end of my presentation, I want to thank the European Union for funding of MAGE, and also the entire staff of the Department of Breeding and Genetic Resources in Mariensee, and our project partners for the very good cooperation, and all collaborative partners of the MAGE project, and finally you for your attention. Thank you.
Did you simulate
gain from having one more generation of back crossing before doing the intercross?
Maybe this was done in Göttingen. I have to ask Professor Semiana. Did you simulate another back-crossing generation?
No, I think you do need to simulate it. Obviously, you will have a benefit from that, but we needed to squeeze it in the image time scale. We opted for a 5-year project.
So you would have done one extra generation?
Yeah.
That's done. Okay.
Questions? Yeah, long day. So then thanks again. I don't know exactly when we should continue. We have a coffee break. We have a coffee break now. At 5:10. I was supposed to be back at 5:10. Okay, thanks. Welcome back. Please have a seat. So welcome to the last session of today. So we have 1 hour for a couple of presentations. And the first presentation will be by Luis on the role of capacity building and updated FEO guidelines to support e-banking strategy. So Luis, please.
Okay. Thank you, Sipka. I will go briefly through the training program that we have followed in MAGE in the framework of Work Package 7. I've listed here the names of several people who were involved in the training activities. Sorry if I missed anyone. So the objectives of Work Package 7 were to demonstrate the benefits provided by animal gene banks and promote their visibility and transparency, disseminate new knowledge, and strengthen human resources responsible for managing animal genetic resources conservation programs, both in Europe and in third countries. And this is the focus of my talk today, is mostly about the training part. Now, these capacity building programs were designed to strengthen the activity of gene bank managers and other stakeholders involved in managing animal genetic resources in Europe and third countries, as I mentioned, but we had to have in mind that these are very diverse groups and needs in terms of the role that they play. These were either gene bank managers, PhD students, research, and so on. The scientific background that they had, from very highly skilled people to practical technicians, and the geographical location, which also placed the language barrier because the courses had to be in the language that was most comfortable for the people attending the courses. Overall, let me go from what we had planned to what we achieved. Initially, we had planned to have 3 courses in Europe, 2 courses in third countries. By the end, we realized 3 courses in Europe and 5 courses in third countries. So actually, we were able to accomplish more than was planned from the beginning. Just to give you some idea of the various courses that we had in different places, these are some pictures that I'll show later on. The first course took place in Argentina in March 2018. Well, this is in Spanish, but that was the poster that they used, was of course in Spanish because the attendants were Spanish. But I suppose you can understand what the title was. This is the group of people who attended the course. We had 22 participants in situ, 16 from Argentina, from various parts of Argentina, 3 from Peru, 2 from Uruguay, and 1 from Bolivia. They all came to attend the course. And the course took place in Balcarce, which is a small town located here south of Buenos Aires. And the local organizers were INTA, a partner in IMAGE, through Maria Rosa Lanori, who is here somewhere, right there, and Lilia Melushi, who is not here. We had 3 lectures from different IMAGE groups, and all the lectures were in Spanish. In addition, we had also the possibility of providing Skype talks to people in other places. So there we had a group of people, 15 people in different places in Colombia who took the course through Skype. It worked relatively well, I think, the Skype part. In addition, we had a field trip to see Creole cattle in the region, and the stakeholders meeting with participants, mostly Creole cattle breeders, 32 participants in the stakeholders meeting. Now, the second course was in Colombia in October of 2018 on conservation and management of animal genetic resources. And this is the poster that they developed for the course. So now the attendants were in place. We had 33 from AgroSavia, which is a partner in this project, and the universities and so on, 33, and then we had 7 students attending the classes by Skype in Argentina. We had 3+1 lecturers from MAGE. One was Paul Butcher by Skype. He wasn't physically there. And these were the 3 participants, and all the classes again were in Spanish. This is the group of participants. The course took place in Bogota, the capital, and then we had a field trip then the last day to Villavicencio, where they have native pigs and Creole pigs and cattle and so on. Now, this is a brief outline of the course. I'm not going to go through the whole list, just you can see the topics are basically the topics of how you manage, how do you characterize and how you manage genetic diversity in the framework of conservation programs. And the course ended, like the one in Argentina, this one also ended with presentations by participants. I'll give you a brief overview of what happened in this presentation, because I think it was extremely useful, this exercise. Now, the first course in Europe was immediately after this one in Colombia, and it was in Wageningen. The first interactive postgraduate course on characterization, management, and exploitation of genomic diversity of animals, October 2018, 28 participants. The picture is a little bit on the dark side, but that's what we have, so we don't really recognize many faces, but we assume that we know who they are. These are the participants from different countries, several countries represented, and this is the list of lecturers who were involved in the course in Wageningen, the first course in Wageningen. And this course received the education prize of the Wageningen Graduate School of Animal Sciences. It was considered the best course as far as I can understand. So they deserve to be congratulated on that. Okay, this is the outline of the course, the topics covered in the course. So you recognize, I mean, the different topics are all related with characterization and conservation of animal genetic resources. And then in 2019, we had the course in France, organized by AgroParisTech in November. And it was a training session for population managers and users of gene banks. And it was organized, as far as I understand, in the framework of the 3rd International Seminar of CRB Anim. C'est ça? No? It was independent. Okay, sorry. Okay, so the trainees were 15 researchers, of which 11 came from different countries. I'm not going to list them. I'll show you a map at the end. And we have a very nice coverage of the world with the different countries included. The trainers were from AgroParisTech, Institut du Port, Institut de l'Élevage, and Göttingen University. The course outline, you can see here the 3 major topics of the course. This course was, of course, shorter than the other courses. The other one were mostly weekly courses, weekly courses, and this one was for 2 days only. Then this course was quite successful, so they decided to have a second course by the end of last year, in December of 2019, in Wageningen again, on characterization, management, and exploitation of genomic diversity of animals. The picture is quite a bit better than the other one. We can at least recognize the faces. trainees 27 from different countries, very diverse group, and the trainers were essentially the same as on the first course. The program outline is, I think, very close to the first one. And then we also had a course in Egypt by the end of 2019, in November, on management of livestock and poultry genetic resources within the scope of climate change. It was organized by the Animal Health Research Institute in Egypt, and the course was, as far as I understand, was in English and some classes in Arabic also. This is the group of students that attended the course, and these are some of the classes. You see our moderator here teaching a class. In Egypt, there were 25 trainees from Egypt, and they were mostly professionals and PhD students. And the course outline you can see here listed. I mean, it's mostly, again, characterization and conservation of animal genetic resources. And then, as well as in Wageningen, the first course was successful, so they decided to have a second one. Also in Colombia, the first course was quite successful, so they decided also to organize a second one. This time, there were 40 students. Most of them were different from the first course, but some were common to the first course. And this was also provided by live streaming to Cuba, Argentina, and Spain. And it worked quite well. The live streaming system worked quite well. The name of the course was Genomics and its Application in Conservation and Characterization of Zoogenetic Resources. The trainers were from MAGE, University of Lisbon, University Complutense de Madrid, and INRA. It changed the name already at this time. It was January, so it should be INRA. Sorry. Didn't update that. The second course, the outline is a bit different from the first one. These are taken from the students' presentation. So they used either their own data or publicly available data sets, so they used that for analysis and for their own presentations. And this was, again, a very interesting experience. Probably you cannot see the titles of the presentations, but there were some very interesting presentations by the students. All of them presented very nice work, and actually we are trying to organize a session within the Congress of the Latin American Animal Production Association to have them presenting their work in this session of the Michel Nav is trying to lobby for that. So let's hope he will accomplish that. Last course was in Morocco, just a couple of weeks ago. Was in January 2020. Biobanking for a sustainable management of farm animal genetic resources, novel approaches in genomics, reproductive technology, and economics. And also there was a stakeholders meeting about cryopreservation of animal genetic resources. The languages of the course here are listed in English and French, but I think also some classes were in Arabic, I understood. No? Just French and English, okay, sorry. Okay, so this is the group of trainees in Morocco, 20 from Morocco, Tunisia, Nigeria, and Sudan, and the trainers were from INRA Maroc, INRA France. This one is updated, okay. Wageningen and the University of Edinburgh. This is the outline of the course with some differences, but it's basically the same framework for all the courses. And so what have we accomplished? Just to summarize, how far were we able to go? I think that the first thing that we noticed was that there is a major demand for training in these areas. I mean, people really need and show the need for the training in these areas. First one. And I think that IMAGE was able to successfully organize various training activities. Usually these were one-week courses with high quality standards. and we made an effort to be in a language best fit to each group. Involved lecturers and students from several MAGE partners, and aimed to build capacity in conservation and management of animal genetic resources using novel technologies. And I think we did quite a nice job in this area. So to summarize, we were able to develop courses in eight courses altogether in 6 different countries, 3 continents, Africa, Europe, and South America. We trained nearly 240 students in these courses, representing 36 different countries. So if we draw a map of where the students came from, someone told me this looks like the coronavirus expansion map, but it's not that bad, really. So we really, I mean, some countries were represented only by 1 student, China, for example, but we covered a large part of the globe was covered by these courses. So I think we can be proud of what we accomplished. Now, I'm going to take 1 minute just to summarize the activities of one of our partners, EFAB, on technology transfer to stakeholders. And their activities have been mostly in developing brochures and newsletters to be used for passing the message to the general public. So we have the brochure, 3 newsletters. We still have 1 newsletter to come. Correct, Duru? Yes.
Yes, this is actually since March 2019. We actually
Oh, okay. So it's more. Yeah, you're right. You're right. Sorry. It's for a shorter period of time. Yeah. So we have more newsletters than I listed here. Program flyers for EAP 2019. Image demonstration desk in the meeting in the EFAB meeting in Dublin 2019. Policy brief and an interview with our boss, which is available on the web page. If you have not seen, you can go there and see. And you have here the contacts of the 2 people who have been mostly involved in Image, Shala and Duru. So if you need anything, you can contact them. Now, I'll give the floor to, sorry, I'm supposed to give this message, remind you that you don't want to miss this meeting in Porto, Portugal, in August, September, first week of September. It's a very nice place. I'm sorry that Andrea Rossati is not here. He would be glad to see this, but anyway. So I'm supposed to give this reminder to attend the meeting. Okay? And now I'll pass the floor to our colleagues from NordGen, who will give you a brief update on the guidelines and what we will have to accomplish in the next 3 weeks.
3
right?
weeks.
Isn't it?
Next week.
Next week?
1 week. Okay. Okay, thank you, Luis. Just a few words about the guidelines. I think most of you that are here were here yesterday as well. Maybe this is the final deliverable from the project. We promised to deliver some guidelines as a part of Work Package 7. Just to tell you how we went about this. As it said in the title of Luis' presentation, it's basically to update the existing FAO manual on cryopreservation. So this is the one that we have used as a basis for this work. But in Image, we also conducted a gap analysis, a quite thorough gap analysis covering lots of details. We did a brainstorm as well in Work Package 2, so that is the other part of the basis for this work. And the third part is that we have interviewed all the work package leaders about the deliverables, the results from their work packages, how they were seeing them fit into the actual guidelines. And as we agreed before, when we started this work, we basically asked 2 questions. What is new, and what should be revised that is already in the FAO manual? So this is basically what it looks like. On the left side is the FAO manual, the outline of how that is constructed. It has 12 sections covering basically everything to do with cryoconservation and a little bit more. On the right side, you can see the gap analysis as we did it in the MAGE project, different headings, but basically we covered the same ground. So now it's up to us in the guidelines to gather all this in a meaningful set of recommendations. I will show you one example, because this is very much a work in progress. This is only started in autumn last year, but only really started here in January. An example from section 5, we suggest a new topic that's called quality management systems. We briefly mention what we actually did in work package 2, what is the basis of any recommendations that we're going to make. I just listed a few bullets, what came out of the surveys that we did. Based on this and based on any other experience we have from this work package, we will then formulate some guidelines. I have given some examples here that what the guidelines could include, and then at the bottom you can see the gap analysis results, just to see if we cover everything that we already pointed at in the gap analysis. So in this way, and this is an outline, we will try to construct the guidelines as suggestions for an update. As we also talked about yesterday, there are parts of the FAO manual that need revision or update that we did not work on in the image project. Those we will not cover in detail. We will just suggest to the FAO that this and this section needs an update or a revision. So in our own guidelines, we will focus on the work that we did ourselves. Now, as you saw that the FAO manual has 12 sections, I will not take you through all 12 of them. Those of you who were yesterday know why. It takes a lot of time, but the only thing I want to say about this is that we have approximately a week and a half to gather all your input and to come up with a draft of guidelines that is a little bit more than bullets. So those of you that have to contribute, be prepared. Thank you.
Thank you. So thank you, Louise and Jaap. With time for one question. It was a nice overview of all the activities in terms of capacity building, training, dissemination. If no questions, thanks again. Continue. So our next speaker is Manuel Luque from Spain. As you know, EFAB, the European Forum of Animal Breeders, is a consortium member of MAGE, and we thought it would be nice to have a member of EFAB, in this case FIGAS, and Manuel is the director of the Federation of Breed Associations in Spain here, and member of EFAB, and we asked Manuel to give some perspectives from the breeding industry on the results of IMAGE or on the IMAGE project. So Manuel,
okay. Good afternoon, everybody. Thank you very much for the introduction. First of all, I would like to express my gratitude for the invitation for taking part of this important event on animal genetic management at European level, and I would also like to congratulate all the IMAGE members project for this important project and for this or for organize this meeting. Well, as you can see in the title of my presentation, I'm going to give you the sector's opinion about the ex situ conservation, apart from some extra and interesting information about the situation of the ex situ conservation in our country. Well, this is the content that I am going to cover in my presentation. Firstly, I would kindly like to take a minute to talk to you about our organization, FEAGAS. And secondly, I would also like to give you some information about the ex situ conservation in Spain. And to finish my presentation, I want to give you the sector's opinion regarding this important topic that we are discussing here today. Well, let's start with the first point. What is FEAGAS? FEAGAS, this word is the acronym in Spanish of Spanish Federation of Livestock Purebred Associations. It was founded in 1982 as a technical and representative national forum for member organizations. It is currently the largest umbrella organization regarding the livestock purebred in Spain. And FEAGAS nowadays groups 113 Spanish purebred livestock organizations belonging to the different species with interest in animal production in our country: cattle, sheep, goats, horses, etc. All of them officially recognized by the Ministry of Agriculture or the different Department of Agriculture of the different regions in which Spain is divided, all of them to manage and coordinate actions related to the herd books and with the development of the different breeding programs in Spain of the different breeds under their control. Apart from many others, our main functions and responsibility are the preservation, improvement and sustainable use of the livestock breeds in Spain. We also represent, manage and defend the interests of its member organization in Spain and abroad. And we also collaborate with public bodies, institutions, university, research centers, and so on, in all actions of interest related with conservation, breeding, management, and sustainable use of the animal genetic resources in Spain. This is basically our organizational structure. On the one hand, we have the typical or the normal governing bodies of this kind of organizations, and on the other hand, we have different technical working groups, but I would like to highlight these two. One technical committee focused on the local breed at risk with different representatives for each species, and other technical committee for the bigger breeds. I mean those breeds, those local breeds not at risk, that we call in Spain the promoted breeds, and also for the transboundary or the exotic breeds officially recognized here in Spain by the Ministry of Agriculture, and with all of them we have a breeding program in place. This is the distribution of the percentage of the different breeds within FEAGAS. As you can see, the highest percentage belong to the sheep breed, and the second higher percentage belong to the cattle species. Well, after this short presentation about ourselves, I would like to go to the second point of my presentation and give you some information regarding the ex situ conservation in our country. Let's start with the historical overview of the gene bank development in our country. And in this case, I would like to highlight that originally, we have paid a lot of attention to all actions related to the in situ conservation, but however, after some years, breeders' associations have paid a lot of attention to the breed characterization because of all FAO recommendations some years ago, and also they have paid a lot of information not only to in situ conservation but also to ex situ conservation as well over the last decades. All this has been the origin of the different gene banks for the different local breeds in Spain, which are located in 3 different locations. We have the CENSIRAS, which is the acronym in Spanish of Animal Breeding and Reproduction Centers, the research institutions, and the universities. All these gene banks have been coordinated by the Ministry of Agriculture with a very clear objective to try to coordinate all this information regarding all these gene banks for different breeds around the country, and mainly with a very clear goal, trying to set up and strengthen the national gene bank in order to store a copy of the different breeds copied in Spain. This is an institutional organization of the animal genetic resources in our country. We have 2 levels. We have the regional governments. In Spain, we have 17 regional governments. These are in charge of the regulation of the smaller breeds. I mean those breeds which are mainly in one region of the country, and on the other hand we have the central government, in this case the Ministry of Agriculture, which is in charge of the regulation of the different breeds located in more than one region in our country. These are the objectives of the national policies of cryoconservation. We manage everything based on this national program for the conservation, improvement and promotion of the livestock breeds. This was born from a royal decree in 2008, but based in all the recommendation from FAO after launching and the implementation of the global plan of action, and after that royal decree was updated last year because of the new European animal production regulation. This is the basis to set up our breed conservation programs, and to know very well what kind of tissues it is necessary to store, the minimum of doses, number of donors, and how to proceed with a copy of the National Gene Bank. On the other hand, we have the how to manage or how to proceed with the task group on cryoconservation and how coordinate all these actions, how to analyze it, and what is the criteria to create and manage all these bank coordinated different stakeholders, as we can see here in this slide. In this slide we can see the involvement of different stakeholders. It started from the breeders associations with a clear idea from them to conserve a breed from these main ideas that I explained earlier, in trying to characterize in a very deep way they breed, as FAO recommended years ago, and putting in place an in situ and also ex situ activities. In that case, we have on the one hand the administration in order to carry out the verification and approval of the breed conservation programs, in order to provide facilities for the gene banks, in this case for the animal breeding and reproductive centers, and also providing subsidies or financial support for the conservation of the genetic material to the breeders' associations, and also to verify the implementation of the breed conservation program through the breed inspector. On the other hand, we have the experts in animal genetics, which help and support all this process to the breeders' associations, and all this information and genetic material go as well to the National Gene Bank in order to store a backup or a copy of the different gene banks that we have all over the country for the different breeds. Regarding the legal aspects, each gene bank establishes its own rules to regulate the ownership and realize the gene plasm. All this, as I explained before, go to the National Gene Bank, which is located in a village here in Madrid, close to here, which is where is the location where the genetic material is stored, owned by the breeders' associations. The stored genetic material can only be used for breed destruction, introduction of genetic variability into already established populations, recovery of the gene bank of the breeders' associations in case of destruction, or the release of germ plasm has to be approved by the National Advisory Committee. The results of all this management and working with information and working with genetic materials. According to the information we get from the Domestic Animal Diversity Information System from FAO, we can compare the situation of Spain compared to the average situation of the rest of the countries in the Europe and the Caucasus region. If we take into account in the left side of the screen, we can see the information taking into account the information of all species. We can see here the amount of conservation material, if we have sufficient or we have enough genetic material stored to finish the gene bank for one breed. Is the green color, orange color. We have started to work on it, but without enough genetic material. In red, with no material. In red, with no information. We can see what is the situation, taking into account all species in the left side, and if we take into account only the main species with interest in animal production in our country, cattle, sheep, goat, horses, and pigs, we can see the right side of the screen, and we can see how we have started to work and to store genetic material for more than or over the 50% of the breed of these species in our country compared to the rest of Europe. Here we can provide you with more details going into different species. In this case is the case of the cattle species, and we can see that we have finished or we have completed almost the 60% of the gene banks of all the cattle breeds in our country, and we have started to work with over the 42 or with over the 40% of them. And I provide you with more extra information regarding the number of embryos, male donors, semen samples, and so on. Regarding the situation of the sheep species, we can see that we have started to work with the 60% of the breed, and we have finished almost the gene banks of the 30% of our breeds. The situation is a little bit worse with the goat species, but anyway, we have finished almost the 20% of the Spanish goat breeds, and we have started to work with the rest of them, which are going on with the storage of the different material or genetic material of these breeds. This is the situation of the pig species, and this is the current doses at the National Gene Bank, doing a comparison of 2018 with 2019, and you can see how the trend is very positive in all species, less in the case of the horse species, where the situation is the same in these both years analyzed. Well, this is the last part of my presentation. What is the sector's opinion regarding the ex situ conservation after seeing all this analysis of the situation in our country? Well, first I'm going to give you the opinion or the ideas of the local breeders associations. Associations believe that it is essential to have a complete gene bank for the breeders. The initiative to carry out the gene banks arising from them are the breeders, following the recommendations of the FAO and administrations. They also tell us that most of the associations think that it is very important to have a backup or security copy of the National Gene Bank, particularly if they are going to face any big problem in the future with the breeds. The ones that haven't got sufficient copy think that they will get in a short or mid-term. It depends of the resources and the problems that they are facing, but this is what they have in mind. The problems that they face, the main problems are the financial support. They receive financial support from the public administration in Spain to do that anyway, because of the huge number of breeds that we have in Spain, and because the situation of the different breeds is very different in Spain, because most of the local breeds in Spain are in danger of extinction, and many of them have not a lot of resources. They obviously ask more financial support to trying to go more quickly with all this process. In other case, they point out the lack of agreement, because they also explain us that they face some problems when the breeders associations have different criteria than the university which are working with the breeds, or maybe with the local administration from which they need to have any support, and so on. They also point out the animal management, because most of these breeds are very rustic, and they also highlight that they face some problems in order to take all this genetic material, because all of the behavior of most of these breeds, and they also highlight the problems to get the samples fertility, for example, in the donkey species. The opinion of the transboundary breeds officially recognized here in Spain is that they don't think it is essential to set up a gene bank for the breeds, particularly in the case of the global breeds, apart from many other things, because of the quick genetic evolution of this kind of breeds. And they agree that the setting up of the gene bank for the breeds is planned because of the recommendation of the administration, and not because because they have a real interest on it. Like conclusions of these three parts, I could say Spain is one of the countries with the highest biodiversity. Over the 80% of the breeds recognized in Spain are local breeds. The breeder associations of local breeds or autochthonous breeds promote the storage of genetic material for their breeds. The Spanish sector believes in the importance of the ex situ conservation, and over the 50% of the Spanish cattle breeds have sufficient genetic material stored, and the rest is in process to be completed by breeders associations. And with this idea, I finished my presentation. Thank you very much for your attention.
Thank you very much for the very interesting presentation and for presenting the views of the breeding sector here in Spain. I can imagine there are some questions from Spain.
Yes. Thank you for the presentation. Most of the breed societies in Spain believe in the importance of having the conservation ex situ and the gene banks. Which are the main problems that the breed societies find to complete the gene banks, besides the money? Because of course, I know that one of the most important problems is to get money to develop these actions. But do you find other type of problems to look for a solution to complete the gene banks?
Maybe this is not a problem of a direct finance, but you have to take into account that most of the local breeds are breeds at risk, and their resources are very limited, and then there are many other priorities. They think that this is important, and they want to continue providing genetic material in order to have this goal in their mind. But you know that the situation with this breed, they have maybe in some cases 1 or 2 technicians to do everything, and they want to do that, but they are facing maybe many structural problems, I think. not direct financial problems, but indirect financial problems.
Lack of resources.
Yes, yes.
Alessandra.
Of course, my question is about the possibility and the willingness of submitting data, information about samples, and in that case, who's in charge, basically, and who should we contact to centralize the effort?
Okay, thank you.
Good to say that also in the room is Fernando Tejerina. Fernando is chairing the network of gene banks actually in Europe, the working group of ERFP. I think you're also in charge for coordinating from a ministry point of view, the gene banks here, right?
Yes. The problem with information is that this information is in the gene bank. The gene banks are in the region, and obviously they are the competence in order to send the information. We have a database that is called ARCA, and in ARCA we collect information from the gene banks, but it's not individual information. It's information as a whole. It's the same information as the gene bank sent to Eugenia. Obviously, I think my opinion, we have to work in two steps, two phases. The first is collect information as a whole of the gene bank, information of the gene parameters, the number of semen doses, the number of males, and then it depends on its gene bank. Its gene bank then can send more information in detail to the MAGE. But I think a good option for the future, we are working also in the working group in the development of a new software for gene banks. Try to integrate the tool in this new software for gene banks to get the possibility to the gene banks to send automatically the information to the European databases, Eugenia and other ones, with the same management system.
Okay, I'm afraid we have to move on. Thank you again for making time. You are a very busy person, so thank you very much. You are very
Thank you.
Give your presentation. The next presentation is Elizabeth, let's go. Presentation about the public-private partnership project on protein markers in goat semen. So Elizabeth, the floor is yours.
So the presentation. Okay. So this is just to give you an example of how in image we could at a moment find subjects that have interested, of course, scientists, me for example, but also a private company and a technical institute in France. So, Okay. So in the course, in the work package 3, we developed an approach, a proteomic approach, to make what we call a phenotyping of the semen, to have an idea of the identity of the semen through the composition of protein of the sperm cells. And this tool has been successfully developed in the chicken, since in the chicken you can have, not so easily, but you can have an access to fertility in very standardized conditions. And also we can get quite a lot of animals. But at the moment we thought that it could be very interesting
to develop the same approach in one mammalian species of interest. And so we looked for which company could have some elements to do that. And we knew that the company named Capgen, in France, involved in the genetic of goat species, could have such information. So the information that we needed were to know the semen, to have semen samples, and to know which progeny have been produced with such a semen. And so after, with this tool, we can study the semen and see the profile of protein, and after that, see if there is a relationship between the protein profile and the fertility, or better, the progeny obtained with the same semen. So Capgen was okay also to do that, but we had to manage that, and we had a partner of IMAGE, IDEL, represented here by Coralie, for example. That is a technical institute involved in the relationship between the scientists and also the different companies. And so it seems that this project has been interesting for the three partners, IDEL, that gave a manager, could pay a manager for this project, Capgen, that so the private companies that accepted to give the information on the semen of the males of Capgen and the fertility results, and so we could start this collaboration this year. It's in the last year of IMAGE. Well, it was in 2019. So in 6 months ago we started. Okay, so you have all that. And so the aim is the identification of new quality fertility and fertility markers of goat semen in order to better predict the fertilizing ability of the semen collection stored in the cryobank. This is the objective inside IMAGE. But you understand that this aim may be applied also to other application in breeding. So the three partners involved are Capgen, IDEL, and INRAE, and we based our work on the proteomic tools that have been developed in IMAGE for chicken semen. Okay, so what about the animals that we could get for that? Oh, okay, not only the animals, human first. First, we needed a manager, and so IDEL provided a manager of the project, a methodology of work that was provided by INRAE mainly, but not only, in collaboration with the two other partners, and the biological material that was provided by Capgen. And so the biological material, it was cryopreserved goat semen, or bug semen, from two breeds, the Alpine and the Saanen, so two breeds in France, okay? And Coralie is a specialist of these breeds, we already had some information from the semen because we got the viability parameters of the semen, the motility parameters also, and more important, we know, yes, the number of progeny obtained per ejaculate. Here, the number of progeny, it is not fertility, but in the first approximation, it is used here as a parameter that is not so far from fertility. After that in the future, probably we will refine the system and work on the echographic images during the gestation. So, which I would say, tools, proteomic tools. So 2 different tools we use every time. On the left, that we call intact cell Maldittov mass spectrometry. That is a quite simple method where you put a cell, here a sperm, or many sperm, on a specific plaque. And after a process, you can get different peaks that show you the peptides and the protein that are in the range between 1,000 and 20,000 Daltons. So in this area, you have small proteins and you have also peptides, original peptides, but also peptides of degradation. So it's a good place to have an overview of protein and metabolic aspects for that. And the other method is a more classic method of proteomic, but also more difficult to get. That is what we call a bottom-up proteomic, because we start by, well, okay, I don't go inside the technique. So you know 2 different methods. And with that, we make 2 different systems of profiling of the semen. And after that, we make the relationship with what we call the fertility. And in a future action, we will use a modeling for all these tools to get a prediction. But we are not yet at this stage. Just what we've seen first, that there were a very high variability between ejaculates. Okay, so at first we had decided to go on a separation between male, but we see we have first obliged to see a separation between ejaculate, even of same male. We selected the good and bad ejaculates, different, and we made the same work on the two breeds, and then we made the proteomic analysis. So what about the first results for what we call intact cell Maldittov mass spectrometry? You see that the alpine, with the alpine breed, you have 191 peaks that were obtained, with the cyanine approximately the same number, and we see that the 2 profiles are not so different, because there are 86% of proximity of the profile between the 2 breeds. This is very interesting, because in the future we will probably be able to join these 2 breeds in order to make a same modeling. Okay, and at this moment, for this method, we have peaks, and now we will go ahead with the identification of all these peaks and with their differences regarding the fertility. Already, we could identify 23 of them that are differentially expressed depending on the fertility. So it's interesting. For the second method, we selected, because it's much more difficult and much more expensive, we selected only 5 good and 5 bad ejaculates. And we made the work, you know, after electrophoresis, etc., etc. And so with this tool, we identify more than 60 proteins, and with 19 of them that are differentially expressed depending on the fertility. So again, now we have another work to do in order to really identify them. So already we know that some of them are already fertility markers known in bovine, for example, or in human. And we know also that some others are not already found as fertility markers. So there is an original part for that. So in the future, we will go ahead with these 2 methods to make them telling all what they can tell, and in order to identify new markers, certainly, in order also to join this method in order to make a modeling for the fertility of the semen of goats and of books. And this modeling will be a great help in order to be more sure that we keep in a collection for the cryobank semen that are much more good than bad. That's all for me, I think. Thank you for your attention.
Okay, thank you, Elizabeth. One question. Beate?
These good or bad ejaculates, were they from different bugs? Or did you have a good and a bad ejaculate of the same bug?
This is a problem at the moment, you know, with the population that we got. It seems that the variability inside the book, in the first approach with these animals, seems to be higher than the variability between bulls. Only for one of the breed, you know, we had no time yet to work on the second. This is what you... No. And so do you have an idea why? Because you know, we have not that in chicken also. The variability, individual variability is usually lower than the variability between males. So this is a question. Probably we will have some indication of answer with these markers, you know, that will be helpful for the future.
Okay, thank you again, Elizabeth. We are running out of time, but I have still one topic to present. So the idea was to be invited colleagues from the UK to present the Generous Bridge project. And one of the main objectives of the Generous Bridge project is to develop a European integrated strategy for plant, animal and forest genetic resources at European level. And our colleagues were not able to come, so my name is also there. I received a few slides. I will quickly go through these slides and try to explain to you what it is about and what we also ask from you as interested people from MAGE and stakeholders in this domain of conservation and sustainable use of animal genetic resources. So, for those who don't know, there are 3 European networks of national coordinators for plants, animals and forest genetic resources. These are networks of national coordinators, and the objectives of the networks are in fact to develop and to implement a more rational system of conservation at European level, to exchange knowledge and information, to collaborate at technical level, and also with a view to share responsibilities and tasks at European level. And this Generous Bridge project has a limited number of partners, but the animal genetic resources domain, ERFP, is represented by, I believe, 6 partners in the project, and the same is the case for the other 2 networks, forest and plants. So the context of this Generous Bridge project, it's a coordination and support action. It's not a research project. It came from a call more than 1 year ago. The project started last year. And I want to highlight the bold text in the call text. It is about joining forces between plant, animal and forest networks to develop and implement ambitious approaches and strategies for the management of crop, forest and animal genetic resources. And secondly, to cooperate between the different communities, to foster genetic resources conservation, also within a wider agrobiodiversity context. So it's not just the pillars plant, animal and forest, but also looking at the wider link to the wider agrobiodiversity context. So why is the European Commission asking for this?
Well, in fact, there is not a coordinated approach at European level, at EU level, when we speak about agrobiodiversity. There's a lot of, there are a number of DGs in the European Commission that have some responsibility for some aspects of genetic resources. DG Environment, the Convention on Biological Diversity and the Nagoya Protocol, DG Agriculture, DG Sante for seed legislation, but also for animal breeding legislation, and DG Research. And there's a number of areas, policies and strategy areas, that could maybe have some better coordination. And the idea is to develop a stronger agrobiodiversity strategies for plant, animals, and forest at European level. And the MAGE project is a research project that has almost ended. Is important to feed into this process of generous bridge. So the outcome of IMAGE is important to develop this European strategy for animals, but also the integrated strategies for plant, animal, and forest. So the expectation of the European Commission for this integrated European strategy is that there will be individual domain-specific roadmaps or strategies. There will be more cooperation between the various networks where possible, and to develop an integrated, wider agrobiodiversity strategy. And what's important to realize that we have a new European Green Deal that has been launched recently. The European biodiversity strategy, so not agrobiodiversity, but biodiversity strategy that we have for some years is being revised at the moment and will be concluded in this year also as a new biodiversity strategy. And there's also an EU forestry strategy under development. So there's a lot of things going on at European level that is important also for us as genetic resources networks to link to or to be part of, and to develop an integrated genetic resources strategy under these broader umbrellas. So as the authors of the slides put it, this is the draft aim of the integrated strategy. We still need to have a discussion on this. So the aim of the integrated strategy is to enhance the conservation and sustainable use of plant, animal, and forest genetic resources in Europe through cross-sector integration, where appropriate, to achieve management synergies, resource efficiencies, and effective policy implementation with the common objective of securing genetic diversity for the survival of humankind. So this is the overall draft objective of this integrated strategy. And I'm not going to read this. It's too much text. But there's a lot of sub-objectives or objectives behind this general objective we have to take into account. So the reason why you have this on the agenda here is that what I said earlier, IMAGE results, recommendations should feed into this strategy development. So all the recommendations from IMAGE should be taken on board to be part of the integrated strategy. We already had a large stakeholder workshop in October where various stakeholders from a different domain were present, and also these stakeholders presented already their views, and we had some discussion across domains, what are the main issues in an integrated strategy. And also the ERFP structures, European Regional Focal Point, the network of national coordinators and experts, they will contribute in the next 6 months to this integrated strategy development. So this is the process that is ahead of us for the next 6 months. I thought there was another slide, but the idea is that the integrated strategy will be developed this year. So by the end of this year, we will have a draft integrated strategy ready. And it's up to us as an ERFP network and all stakeholders that are present to feed into this strategy development process. So we heard all the recommendations already of IMAGE. We have presentations, we have discussions, but maybe there are some more general recommendations from you now. We do not have a lot of time, but if you want to put something forward now that should be really part of this integrated strategy from an animal perspective, you can also raise it now. And many people who are now in the audience are also involved in the further process through ERFP. So thank you, and maybe there are some suggestions and comments here. Any comments, suggestions? Michel.
I think that from the viewpoint of animal gene banks, it should be relatively easy to identify common steps across domains. For the in situ, I see it much more difficult. Maybe we should focus on what we have developed in image on complementarity between ex situ and in situ. But what I found interesting in the workshop last October that there are similar objectives, similar wording of objectives, but under the objective, people are doing different things. Either because the sociology of the actors is different, or the biology of the species is different. So an integrated strategy would be to agree on the main objectives, but the implementation probably will be different. But we have some lessons to take, at least for gene banks. That's what we see in France, maybe what you see in Netherlands, where you have gene banks for plants, animals, and forests in the same organization. There are some common, reciprocal interest. For example, for the plant gene banks, they are using the resources much more for breeding than what we are doing in animals, where it is for conservation. But in plants, they do not freeze for long term. They have to regenerate every 10 or 15 years, so which is very costly. The advantage is that they check the phenotype, but then they do not conserve more than 10 or 15 generations in animals, because we have the liquid nitrogen. We can conserve for much longer time. But then maybe it's more expensive for animals to take it out and produce animals than for plants. So I think there's this sort of experience that we could share to see whether we can still change or test other methods, really from the viewpoint of gene banks. But I am very surprised that in forest there is a complete separation between the in situ conservation and the gene banks that are for research, only almost, and the in situ, which is for forestry program and almost no research. At least in France, it's completely different governance. Which, when I discovered that, I was really surprised, even shocked, because I think it is more productive to have more exchanges. But apparently, this is the history of the forestry world. I don't know. That's because the trees, they grow for longer, and so it's completely different perception of one individual tree or one sheep. Of course, it's so. I think it is an enriching to discuss, but I don't think we can come to practical recommendations, which are for the three domains. But at least see how we translate a similar objective in different actions.
I think it's very clear that technically the domains are very different. So I fully agree. An important framework we also take for our integrated approach is the FAO framework. So it started with plants and animals, forests, and now FAO is also working on an integrated biodiversity for food and agriculture strategy. And if you look at the higher level, we have more in common than we think. And we should be more visible, this agro-biodiversity domain should be more visible in the larger biodiversity context. And I think that is one of the main objectives here, to see how we can have a better position from this area at European level, and to develop some, I think not at a technical level, but at a more strategic level, where are the common issues and synergies and objectives. Who else? So, what I said before, ERFP is part of the game. There's a group in ERFP who has to run the show to develop the strategic priorities from ERFP, from the animal side, and we take into account what comes from the projects like MAGE and from stakeholders. And in the end, yeah, we have to end up with an approved integrated strategy, approved between the 3 domains of plants, animals, and forests. And it sounds like a long process, but we have only 1 year to do this, so it's quite a challenge. So, if there are no further comments here, I want to give the floor to Michel for closing remarks of the whole session of today.
It has been a long day. So I would like mainly to thank the speakers, because I'm quite happy of all the presentation we had, which I think have really shown the different components of the gene bank, what is the life of a gene bank, and what are the missions, and what are the challenges for gene banks. And in the different work packages, I think that objectives have been reached as compared to what we promised. Of course, we have to wait for the Commission and the experts to agree with us, but I think we have very good examples of the different objectives that we set up at the beginning of the project, but we also identify future work needed, both for the fertility, for also the economic analysis, probably economic analysis, which was really probably the first real organized study at the European scale. And we have a lot of questions to enrich these economic studies. But for molecular tools and breeding tools with the SNP chip and the MOPS program and the portal, I think we will have already solid building blocks to help for the future development of gene banks. And so we have a busy month of February, all the partners, and then we will put on the website of IMAGE more information, because at the end of the project, that's where we finally we have a better vision of what we can make publicly available on the website, as compared to the beginning of the project, where sometimes we say, well, we are not yet sure whether we put this or not on the website. Now we can really provide a lot, I think, on the website. And so if I understand well, Luis considers maintaining the website even after February 29, which is very important, so that people can still go back to this website to find our results and main features or case studies or deliverables. This is important to continue. And there are some other EU projects which I hope will inherit some of the recommendation of IMAGE. And now we have also all of us to to lobby or to send messages back to the EU, so that there is still work to continue for gene banks, and we can also send some maybe precise messages showing what have been done. And I think all the colleagues here have done a lot, and what we would like to continue for a good development of gene banks. So tomorrow for the partners, we will have this real action plan until February 29, all the final version of deliverables and reporting, so the story is not finished for them. And for the, well, if we can get feedback from you or from colleagues, I think it's always possible. We have to organize this maybe on the website to get some contact address. But I would like really to thank all of you and Susanna for the organization, because I think it was probably tiring, but I hope very enriching day for all of you. And so I thank you for staying until the end. Thank you. And so we'll have abstracts. I have asked abstracts to all the speakers. So those who did not send the abstracts to Anthony, please, you send the abstract, because then we will make the abstracts available on the website and also on the deliverable. One page abstract, English. You can put text, picture, but one page abstract. Thank you.